Alright guys, welcome back to another review. So in the last published video, it was biology, and now I'm doing biochemistry. Now, the thing about it is, you'll notice we're not on chapter 1 biochemistry is because there's some things in biology and biochemistry that are important to go over, and a lot of the pathways that are important in biochemistry were listed in the appendix of the review sheets. So you want to go to the PDF and scroll down or go to the bookmarks tab of the PDF. You could use any PDF viewer and they should have that, be able to open that up for you. And then you basically just go and find what you're looking for, right? So go to the appendix. And you notice that Appendix A is actually organic chemistry, so we're not going to do that. But biochemistry is really related to biology, right? So what I'm going to do is go over the heart, oxygen transport, and then brain, and then endocrine, and then afterward, all of this stuff down here, such as DNA, RNA, DNA replication, the central dogma, amino acids, enzyme, and everything down here is basically all the biochemistry stuff and pathway. So this is why I'm highlighting the appendix now, because it's very important for you to learn the biochemistry. So as we go through Biochem 1 and stuff, you'll see that. But for now, we're just going to go through this part of the appendix that's more so related to biology. But you're going to have to learn this anyway, for the sake of the test anyway, so it's really important to learn. So let's just start with the heart pathway and some parts of it. So you see here the anatomy of the heart. Here you have your aorta. It's this big artery, right? And then you also have your pulmonary artery here. This is your pulmonary artery. And then it's harder to see in dark, so I'm going to try to put it in light mode today. But as you can see, you have your right atrium over here and your left atrium over here. Now I used to be confused, like, why do we have sorry, this is your left atrium here. I used to be confused why the left atrium and right atrium were not named like left and right, right? This says right atrium, but it's on the left side of my page. Or right atrium, and it's on the left side of my page. And the reason for that is because, sorry, let me get the pen out. The reason for that is because it's a reflected view. So it's your left, but it's the patient's or whoever's heart that you're looking at. It's their right. That's their right side of their heart. All right. So yeah. And then you also have these ventricles. Here's the left ventricle. The right ventricle. Even I'm getting confused. And the left ventricle is on the right side. Okay, it's confusing me already. And then you also have your valves that connect. So the avioventricular valve connects the atrium to this right ventricle. So right atrium and right ventricle, in between them is the avioventricular valve. And then for the left atrium and the, where is it? The left atrium and the left ventricle are connected via this atrioventricular valve as well. So that's easy. It just mirrors each other. And now it's hard to see a little bit. Let's do like a purple. When the blood flows upward to then go from the left ventricle to over here, you get, where's this, left ventricle over up, right? You do have a valve, and it's called the semilunar valve. When you go from the right ventricle and you're moving up over here, there's a valve there, and that is called also the semilunar valve. And then so you can think of the heart as like, yeah, like blue and red. It's not actually blue and red, and it kind of mirrors each other in a way, except this aorta is so big, by the way. And then studying the blood pathway, so I have some biology video on it already, so we don't have to go over that again, which is great. And then how does your blood flow through the heart and your overall circulatory system in your body? What does that look like? Well, it looks like, well, what is the pulmonary valve doing? So you start with 1, right? You start in this right ventricle, and you just follow this path literally. So 2, practice naming it yourself. So this should be your pulmonary pulmonary vein, I think. Why is it not there? There's no label for 2. That's so weird. Pulmonary artery, sorry. It's going up to the pulmonary artery. Let's check that's right. Yeah, this is called the pulmonary artery here. So 2 is the pulmonary artery. And by the way, it has to pass the valves, right, as well. So pulmonary artery, and then it goes to capillaries of the lungs, so 3, right? And that's where your diffusion of the gases happen, diffusion and exchange, basically. And then you should get down and follow through. I'm trying my best to follow through with this because other people should be able to follow through as well. But this is basically 3, and then 4 is when you go down and then into here, which is your left atrium, and then down, and then down to your this area here, right, which is your ventricle, right? And then 6, and then it's bringing blood back out, and then through the, now it's too crowded here, sorry, to the aorta. So it should be your aorta bringing this out. Now it's going back to your body, right? It's going back to your body, back to your body. And I believe this is oxygenated blood back to your body. Let me go check that. So here we go. It says here, most arteries carry oxygenated blood away from the heart to the rest of the body. Alright? So that should be good. Okay, and now we have electrical conduction of the heart as well. So remember the pathway. This is just a diagram for you to help you see it. All of that review was in the biology video. So this is just an image to help you kind of visualize the pathway. Okay, one second. Alright, so now it's getting into more details about what blood actually is. Hemoglobin, in other words. So hemoglobin is just found in blood. Sometimes it's written as HB, I guess. And it has 4 polypeptide chains, each combined with an iron-containing heme group. So this is what it looks like over here. Not the whole thing, just like one of them. So maybe I'll just circle that. Yeah, so here's iron, right? And so most oxygen transport, that's how your blood is able to transport oxygen. Most oxygen transport takes place through the use of hemoglobin. So hemoglobin is kind of like a carrier for oxygen, right? Because a small amount will still dissolve in the plasma, so in your blood, and be transported on its own. But most of it is transported via the hemoglobin. It's a big molecule-looking thing there. Each RBC contains 2.7 to 10 hemoglobin molecules. So cooperative binding is when O2 binds to one of the 4 binding sites. So I'm going to show a different picture of hemoglobin, because seeing different pictures, different ways people explain things, and doing practice questions help you to see that as well. Seeing the different ways help you understand the material in a deeper level, more than just seeing things in one way. So as you can see here, we have this image, and you can see that it's very it's very like spaghetti-like, and then you have your 4 binding sites here. And so the polypeptide chain, the reason why it looks like spaghetti is because that's one of your proteins that have been wrapped up already, right, into this cell. And this cell is a special cell, right? It doesn't have any organelles, and you remember that from biology that we reviewed in the other video. And so these are your binding sites, right? And the heme is there, and the oxygen can kind of bind there. So that's a simplified look at it. There's other images, lots of them, and each site can have a name as well. I just want to go, as you can see. So this is a good picture. Okay, and so you have cooperative binding, where basically once the O2 binds to one of the 4 binding sites, it becomes more likely that the remaining sites will also bind, so it increases that. So that's why we call it cooperative binding. And then for CO2 and H plus inhibition, allosterically inhibits hemoglobin. That means CO2 and H plus will trigger the heme group to then release its O2. So give me a second. Okay, so here I wanted to provide a more structured lesson to what we're about to talk about for CO2 and H plus inhibition. So remember we just talked about hemoglobin being like a delivery truck of oxygen. So it's able to carry oxygen. Some oxygen will dissolve in your blood, in your plasma. Plasma is like your blood, right? A specific part of your blood. And so that's able to happen. So we can. So you think of it as a delivery truck that loads up oxygen packages, the 4 areas, right? Like it has 4 of those units. How tightly the truck grips on its packages and eagerly it lets go of them is exactly what the oxygen hemoglobin dissociation curve measures. So a right shift means that, so oxygen hemoglobin dissociation. So if we go back down, this is the curve we're talking about here. Okay. So if a right shift happens, the truck grip loosens and it unloads O2 more easily, and that's good for tissues that are working hard and screaming for oxygen. A left shift happens when the truck grip tightens and it holds onto O2 longer, good for loading up the lungs or for fetus stealing oxygen from the mom. Remember when we said that fetus has higher oxygen affinity than their mother. It loves to steal oxygen from their mother. Okay. And so we'll go over the oxygen dissociation curve more so in a second. But basically what you have to get from this is that kind of like hemoglobin can have a scale of grip strength on the oxygen, how tightly it holds onto it can be loose and it can be very strong, right? So when we talk about the cooperative binding and the inhibition, so your working muscles produce CO2, and that CO2 goes into your red blood cells, where carbonic anhydrase, an enzyme, converts it, CO2, so this is your buffer system. This is your buffer system, and I'll write it out better right now. Alright, I didn't really feel like writing out the whole linear equation thing of it, and I also felt like showing a picture would help. So this is one picture showing how the buffer system works. If you learn it this way, I think it helps a lot more than just learning just a linear look at it. But here's a linear one too, or this is a more linear one over here, but it's essentially the same thing. Here we go. CO2 plus H2O leads to the carbonic acid at some point. Let me see, blood buffer system. So your carbonic acid, right, breaks down into the H plus, and then we call this bicarbonate, this HCO3, and then yeah. So I'll put this over here, so maybe it'll be easier to see. I'll put this on a gray background, so it's easier to see. All right, so that's that. And now, so the working muscles, yes, so yeah, and that's the buffer system. And we already reviewed that biology, so you can go back to that video. And so that's like, imagine a delivery truck arriving at two houses. One house is really quiet and one is really loud, right? So one of them, again, is screaming, and the other one is really quiet, right? So the truck instinctively drops its packages at the loud house, or the house in need. It knows it's in need. And that's exactly where the CO2 and H plus build up, active oxygen hungry tissue. So CO2 and H plus build up in that tissue. And in other words, muscles use a lot of cells use a lot of they make a lot of CO2 and H plus, and so they're oxygen hungry. Remember the Le Chatelier's principle. You can the bicarbonate buffer system is all across chemistry. It's talked about in biology and biochemistry. So definitely high yield. Not as much biochemistry. This is more like biology review anyway. So the rule is that when your pH lowers, your heme affinity for O2 lowers. So the curve shifts right. So this curve will shift to the right, or left. That's all you have to understand now. So it will shift to the right when the pH is low and you have low heme affinity. So in MCAT, I don't know why it says hook, you can say mnemonic, is cadet face right. So CO2, acid, DPG, exercise, and temperature. So these are all situations when these increase, these four factors, 1, 2, 3, 4, 5, sorry, five factors, and these go up, any of them go up, they will shift the curve to the right. So you have a lowered affinity for oxygen in these scenarios. Now the right shift, so now the right shift, or lowered heme affinity for oxygen. And then so that means the heme will basically want to give out its oxygen or release it, right? So it has a low grip strength, as we said before. And now the right shift equals lower affinity, so more O2 is unloaded on tissue. As I said, it has like lower affinity to oxygen, so your heme from your hemoglobin is like, all right, I don't want it, take it, and it unloads that O2 to those areas that really want it. And so in times where your body temperature is high, or like think about when you're exercising, right? So you have CO2 and H plus buildup, and then you have that hemoglobin with its 4 units, right? And heme units, and it can then give this one oxygen, the muscle. So I didn't draw the muscle, but that's your muscle, and it'll drop it off, because then at that point it would have had lower affinity to oxygen. And then for 2,3-BPG inhibition, and by the way, we're just basically going over this, but I'm going over it. So this is 2,3 CO2 H plus inhibition. So I'm just explaining that. And so we explained bicarbonate buffer already, and we explained the oxygen affinity here. We explained oxygen affinity from here. We also included this enzyme. And then now we're on this 2,3-BPG inhibition, but I'm adding some more explanation so it's more clear. So the 2,3-BPG inhibition, 2,3-BPG is a second allosteric regulator. So basically, an allosteric regulator is a molecule that binds to a protein or enzyme at a special site called an allosteric site. So you'll notice allosteric more so when you learn about enzymes, but yeah, give me a second. So then basically it wedges itself onto the central cavity of the hemoglobin tetramer, and forces alpha and beta subunits to let go of their O2. Remember that picture I showed you of the subunits of hemoglobin earlier? I have to get it back now. Actually, was it here? No, I didn't save it. Okay, so let me show you hemoglobin again. Where was the subunit picture? Here it is. So here, I'll copy it down. So remember, you have 4 units, and they're just called the alpha, beta. Alpha is here, and then beta is on the side. This is also beta. So alpha. So the subunits are called stuff. And so, yeah, we already went through this. Now we can go through the analogy to help you understand. So let's imagine hemoglobin as a 4-seated car. As we've imagined, it's a carrier, right, of oxygen. So 1, 2, 3, 4. These are the 4 seats. And so now it has 4 seats, right? And the 2,3-BPG climbs in the middle console and shoves out. We're kicking out O2 out of the alpha beta seats. So 2,3-BPG, 2,3-BPG. I just want to show a little diagram too. Okay, I just want to show this diagram. So you can think of 2,3-BPG as a little molecule, right? And so what it's doing is that if it goes into here, it can shove out, kicking O2 out of the alpha beta seats, right? If it climbs in the middle. So then fetal hemoglobin swaps. So for fetuses, for their hemoglobin, it swaps its beta seats for gamma seats, and gamma seats don't have a spot for 2,3-BPG. So what does that mean? That means that that's one of the reasons why the fetuses have higher O2 affinity, because they have gamma seats, not beta seats. And so because they have gamma seats, they don't have a spot for 2,3-BPG to come in and kick out O2. So their O2 would be higher than the mother's, because the mother still has alpha and beta seats, whereas the fetus has gamma instead of beta. Or actually, it's the other way around. whoops. And so the net effect is that the fetus's blood, HbF, the fetus's hemoglobin, sorry, remember I said HB can stand for hemoglobin, ends up holding more O2 than the adult HbA. So you'll note that we reviewed this in biology as well, so we're good with that. So an increase in this will shift the curve right, same direction as CO2H+, so more BPG, your body is signaling that it needs more oxygen delivered. So it's this situation here, like the cadet. Okay, and then now, so we went over 2,3-BPG, so I can check that off. Check, and now we're going into, and we should have went over fetal hemoglobin already. And then I think chronic hypoxia and high altitude are also situations where this happens, where you have this increase in 2,3-BPG that shifts the curve rightward. So more BPG in your body means that you'll have the heme group will have a lower oxygen affinity and want to give that oxygen away because it knows your body needs that oxygen. So hypoxia, when you have oxygen deprivation, so we can check that. And now we are on CO2 transport. So CO2 transport is back to the lungs. So the delivery truck doesn't drive home empty. So not only does your hemoglobin, this guy, have oxygen in it and it drops it off, it will also have, it will also then, afterward, the CO2 that triggered the release stays bound to the hemoglobin. So what it's doing is it drops off loads of oxygen, and picks up CO2 as it goes, right? So let's say it's dropping off to muscles that really need oxygen, you're exercising, and those muscle cells really need O2. So your heme is going to drop those off and then pick up CO2. So it doesn't just drop off, it also picks CO2 up. And when it happens in your lungs, what it's doing is that it loads the truck with oxygen. So from your lungs, you're getting oxygen. And then when you leave your lungs, drops off CO2 it was carrying. Right? So in your lungs, it's picking up oxygen, then dropping off CO2. But in the tissue that needs it, any tissue, like let's say it was the muscle, something like that, your hemoglobin is giving away this oxygen, and then it's taking in instead CO2. So it's giving away oxygen and taking in CO2. So it's opposite there. It's a continuous loop, not two separate errands, right? So it's doing two things at once. Whoops, not like that. So it's doing two things at once. It's focused on the oxygen and the CO2. So it can carry both. That's what your hemoglobin, HB, does. Okay, so then fetal hemoglobin in P50. So hemoglobin and P50, HbF has a higher affinity, as you said, than the adult because gamma subunits don't respond to 2,3-BPG. We already went over that. And the result is the dissociation curve shifts to the left as the BPG levels were low. So at the placenta, the mom's hemoglobin and the fetus hemoglobin are both reaching for the same pool of oxygen. And because the fetus hemoglobin ignores BPG, right, it can hold on tighter, so it has a higher affinity to O2. O2 affinity is higher for, again, it's the same key concept that we're going over again and again. And so it wins the tug-of-war and pulls O2 across the placenta from the mother to the baby. And so P50 is the oxygen pressure at which hemoglobin is 50% saturated. So think of it as a price tag for how hard O2 has to push before a hemoglobin will accept it. So if you have a low P50, it means hemoglobin will bind to O2, so it'll take in O2 easily, even when oxygen pressure is low. So that means you have a high affinity to oxygen. So fetus hemoglobin has a lower P50 than adult for this reason. It's more eager to bind oxygen. So it has a high oxygen affinity, but the P50 is lower because P50 by definition is the oxygen pressure at which hemoglobin is 50% saturated. So this way you can memorize there's a left shift, which is higher affinity, but it lowers P50. You can have a right shift, which lowers your oxygen affinity, but raises P50. So basically what P50 is doing, in other words, is this, I try to give different ways of explaining things because different definitions might connect differently for different people. P50 measures how tightly your blood holds on to oxygen. So normal humans P50 is around 26.6 to 27. I have an HG, here's a content for that. Lower P50 means high affinity and they're holding onto oxygen tightly. A higher P50 means low affinity, letting go of oxygen easily. So there you go. And this is how it relates to the curves. So we went over that one. Now we can go over sickle cell anemia. So we just went over, P50. Good. CO2 transport we went over just then, and then now we're on sickle cell anemia. Okay, so for sickle cell anemia, the molecular cause of it is that a single point of mutation. Remember we learned about point mutations. That was in biology, unit 12. So it's at the very end of the video basically. And beta hemoglobin gene swaps glutamate for valine. So soon, this is why I wanted to do bio with biochemists because these are amino acids, which we'll learn soon in unit 1 of biochemistry. It's in unit 1 of biochemistry or chapter 1 if you're following with the textbook. Right, so for valine, so glutamate, is swapped for valine at position 6. So what the analogy is that glutamate is like a smooth, water-loving puzzle piece edge, so polar and charged. Polar, because water is also polar, so H2 is also polar. Like dissolves in like, and so hydrophilic. Glutamate is hydrophilic, is what we would say in other words. And so it doesn't stick to its neighbor. Valine is greasy and hydrophobic, right? So remember in normal science class, they'll be like, oh, oil doesn't like water and they don't associate with each other. It's kind of what it is. It's hydrophobic. Has a phobic. Doesn't associate with water. Once that, it's like a strip of Velcro. So once that Velcro patch appears on the surface, especially when hemoglobin is deoxygenated, hemoglobin molecules start snagging onto each other, chaining into long, stiff fibers instead of staying separate. So in sickle cell, if you look at sickle cell anemia picture, get one, you end up having a picture that looks like this, where you have this sickle cell instead of a normal red blood cell, right? So now, yeah, you have this long, stiff fibers instead of staying separate. Those rigid fibers physically bend into a crescent or a sickle cell shape. That's why I had to show the picture of the sickle cells. So sickle cells are fragile and get stuck in small vessels, which causes local hypoxia. So this relates to what we just learned about hypoxia over here with 2,3-BPG inhibition over here. So now, and low O2 promotes even more polymerization. That's a vicious cycle behind sickle cell pain crisis. All right. So one wrong amino acid in the chain is enough to change the whole protein's behavior. Sickling is worsened by anything that lowers O2 or decreases deoxy-HB. Dehydration can worsen it, high altitude, strenuous exercise, cold, vasoconstriction. So think of these examples and then think about what's happening behind the scenes in that person's body. So when they're in high altitude, what's happening to all these mechanisms that we were just talking about. All right, now that is all done for this side. We don't need this anymore. So there's also something called myoglobin. We've been talking a lot about hemoglobin, but there's also myoglobin, and as you can guess, it relates to muscle. So it stores and releases oxygen as well. It is a monomer and contains only one heme group. So myoglobin is not pH sensitive. That's why we don't have to talk about the bicarbonate buffer system in this case. So O2 affinity, myoglobin has much higher oxygen affinity than hemoglobin. And that makes sense because your muscles need a lot of oxygen. And this means it can bind more securely for energy. And as you learn soon in biochem pathways, that you need a lot of oxygen to make cellular energy, ATP. And so, but you can ignore what I said then. Just know your muscles need a lot of oxygen to make energy. Heme group. So for the heme group of myoglobin, there's only one, right? And this cannot exhibit cooperative binding. It has a hyperbolic curve. 2,3-BPG has no effect on myoglobin. So as you see, myoglobin is very much unaffected, but it just loves O2. You know, it's staying in its lane to help oxygen get. And then so now we're gonna go quickly over the brain lobe. So you should have done this with psychology. You should have done this study during psychology. So these are just the different parts that you have to know. So front lobe, you have your frontal lobe. Why does it say back here? Oh no, this is just orienting people, sorry. So they're just saying front and back here to say this is the front of your brain of the side and this is the back side of your brain. I thought it was referring to the lobes, it's not. Anyway, these are parts of the limbic system to memorize. So limbic system has different parts. And then you have to know the lobes. So let's just go into the lobes because those are the bigger sections. So parietal lobe, occipital lobe, temporal lobe is there. Where's frontal lobe? Frontal lobe is there. So first you can learn the lobes. That's a big section. It's big sections of the brain, 4 big sections of the brain. And then you can learn more like specific stuff after. So what is the limbic system and what parts are in the limbic system? So limbic system. Let me just show this. So this is the different parts in the limbic system. They include your hippocampus, amygdala, hypothalamus. Oh, what's happening? And cingulate gyrus. So, and there's other supporting structures too. So hypothalamus, amygdala, hippocampus. And then learn everything else here. Cerebellum, brainstem, what is that? So what structures, where is it, and what does it do? So this goes into even more depth of your brain, which you need to know, which during that psychology video should have went over more about the brain, but this is also part of biology. So cerebrum is a higher functioning part. So cerebrum is literally all of this, I'm pretty sure. So cerebrum, I'll just show a picture so it's easier to see. This picture isn't actually that good, this diagram. Okay, my bad. So the cerebrum is not like that. All right, yeah, so it is kind of like this, where, you know, your cerebrum is like this. This is the largest, whoa, what's happening here? This is the largest group you can group your brain into. Like, this is what it looks like. And so this whole part here in pink is your cerebrum, right? So if we go back, where does it say cerebrum? You have your cerebral cortex, prefrontal cortex, part of it, frontal lobe, temporal lobe, parietal lobe, occipital lobe. All of this is under your cerebrum, and they all have different functions. And this is all psychology review, so you should know this. But your cortex, cerebral cortex, has primary and associative cortex, and then everything listed after that is trying to explain the function of what that part is doing, right? What's the point of knowing the part if we don't know the function? So yeah, so you should know the part and the function. Frontal lobe, Broca's area, is known for speech production. It contains Broca's area. So Broca's area is an even smaller area within your frontal lobe that's meant for speech production, right? Wernicke's area is meant for language comprehension, and that's a small area, Wernicke's, inside of your temporal lobe. Temporal lobe as a whole is also going to be processing other stuff as it lists here. Parietal lobe has functions too. Specifically, like a high yield thing to memorize is that parietal lobe is for processing perception of stimuli, and then your occipital lobe is easiest to memorize. It's for visual processing. But all of that should be mostly review. You also have your cerebellum, which was over here. It looks like a tiny brain inside of your brain, and it's called the cerebellum. And it's for movement control, speech control, and any damage to the cerebellum produces disorders in fine movement, equilibrium, posture, and motor learning. So this damage can also affect speech and also eye movement. Yeah, and then for your limbic system, oh yeah, it lists all the parts in the limbic system. Hypothalamus, amygdala, thalamus, hippocampus. So hypothalamus, amygdala, hippocampus, and where's the thalamus? Wow, they didn't list the thalamus. I don't see it here. All right, so just know the thalamus is in there as well. So yeah, and then know the functions. And for the hypothalamus, it may help to remember the 4 F's of the hypothalamus. The 4 F's hypothalamus. I'm pretty sure I can't say one of them on the screen, but these are the 4 F's. Okay, now we can go on to the brainstem, which is literally in the name what you think it is. It's the brainstem. So this is the brainstem, and it's for most of your body's basics. So pons, reticular formation, medulla, and long tracts are all parts of the brainstem, and the pons are responsible for your waking and relaxing. So just like think about sleep, basically, reticular formation, alertness, motivation. Also autonomic functions like circulation, respiration, digestion. Medulla regulates autonomic activity of your heart and lungs, so that's really important to memorize. And the long tracts are collections of axons. Remember, we did that in biology review about neurons on the nervous system chapter, so we can go back to that if you don't know what axons are and neurons. Connecting the cerebrum to the spinal cord, passing through the brainstem, and upper motor neurons signaling down, and somatosensory long tracts signaling up. And then your brain is also made up of your left hemisphere and your right hemisphere, right? So 2 halves of your brain. And so they say usually that your left side is meant for like language, logic, math, reasoning, and positive emotions, happy. And they say the right side is for negative emotions. And they also say it's for creativity on the right side, creativity. So art, music, left-hand control. Oh, left-hand control is also important, and right-hand control. And this is for right-handed control, left-handed control. Now, another thing about the brain to note is that if you notice, the brainstem is all of the basics that your body needs, very basic functions that a lot of other animals have too, right? But as you get more forward and up, let me just remove this, all of this. So from the bottom, right, that would have been our brain, you know, at the very start when we were just looking like chimpanzees, right? We didn't have that much. But then as we evoluted, we started to get all this cerebrum area, right? And this, as you move higher up the brain, you'll notice that we move from basic life functions to more advanced. So you can always think about the other parts being more evoluted, or like more advanced evoluted. And this bottom part, like the brainstem, where that is it's more like, it's more basic, and even the cerebellum is at the bottom, and that's just for like movement and stuff like that, right? So this is very basic stuff, functions, and then all the structures at the top are more like advanced, like thinking, you know, where is it? Prefrontal cortex is good for planning, you know, and the reasoning, planning, speech production, and all this other communication stuff. So that's just one way to think of it. You don't have to think of it that way at all. And then endocrine organs and hormones. This is also part of biology. So basically all of this was already talked about. It's just listed out for you in a more detailed manner. So you can always go over that as part of biology in the endocrine when we're studying the endocrine system there. Okay, and for lab techniques, I'll go over this probably afterwards for biochem. Or I might go over it for organic chem, because organic chem is also related to this. So I'll put a star here just to go back to this later. And then for DNA and RNA, this is important, and we'll go back to this soon. So I'll put a check mark box there once we finish this, because this is all this stuff here. I'll circle it a little bit, because this is all going to be reviewed in biochem right now as soon as we get to that. So even enzyme inhibition is part of biochem, and metabolic review. And then after that, there's just a bunch of metabolic pathways to know and memorize, which is gonna suck a little bit. And then the central equations sheet at the very end, Appendix Q, is for physics. So this can help you as you do physics problems to refer to this sheet. Or you can make your own sheet and know where everything is, and that's probably a better way to study, because you're actively making your own sheet, review sheets, which is good. Okay, so part of Unit 1 Biochem is to review amino acids. So amino acids, there is, well, we can count them together. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. A good 20 that you have to memorize, right? So there's 20 of them, and you want to memorize them by their characteristics here. So, and also what they look like, because on the test they'll literally show images of the amino acid, and you literally have to be like, oh yeah, that's serine, and like just the picture, right? So imagine basically all of this is blocked off. Whoops. Yeah, imagine like all of that is blocked off, and you basically have to memorize, what is that? That's serine. What is that? That's threonine, tyrosine, aspartate, so on. So you have to know that. You also have to know these one-letter abbreviations. So it looks simple for glycine, it's just G, right? But if you go to lysine over here, it's K. If you go to arginine, it's R, which isn't too bad. Glutamine is E. Aspartic acid is D. Glutamine is Q. So as you can see, not all the one-letter names are matching, and it can get kind of confusing. So you also have to be able to memorize that as well. One way I would do it is use like block off these and try to memorize it by name and the 3-letter abbreviation too. So this is the 3-letter abbreviations of each of them. So you have to remember the 3-letter, the 1-letter, and you also have to memorize the full name based off of the image that you saw. And then you have to know that glycine is part of a nonpolar group. So it's nonpolar. Glycine has a characteristic of being nonpolar. And then you have to memorize, for example, serine is polar. For example, aspartic acid has a negative charge. And then positive charge, for example, arginine, right? So you have to memorize, oh, what's all positive charge, right? What's all positive charge? It's going to be lysine, arginine, histidine, right? So yeah. And these are all listed out for you here. Everything you have to memorize actually in this list as well. But you also have to practice knowing the actual drawing diagrams of the amino acids. And this is some extra stuff here to know. Okay. So that's why I introduced the amino acids. You can see appendix for the full AA chart stands for amino acid chart. So we just went over that, which is great. So study that chart. And now we're officially starting Biochem 1 at this point. So amino acids is a molecule with 4 groups attached to a central alpha carbon. So what we were looking at was not necessarily the full amino acid. So I'll show you what, oh, it's over here. Okay, good. I'll show you that this is the amino acid structure. Good, I didn't have to Google a picture. Where you have your carboxylic acid group, you have your amino group. So amino is that NH. Carboxylic acid is the COOH. Right? And then you have this variable R, and then you have this central carbon, right? So this central carbon is connected to, you can think of it as connected to, I don't know why it says 4 groups. Oh yeah, because the hydrogen, sorry, I forgot to add this. Hydrogen is its own group here. So this is the central carbon right there, and it's connected to these different 4 groups around it. So this is connected to hydrogen, the amino group, the carboxylic acid, and the R chain. Now for the R side chain, that's important for what we just reviewed in the full amino acid chart, is because that R side chain is where glycine, for example, would be, or where isoleucine would be. So like, one sec. So a more proper way of how I can explain it is that threonine is not within this R chain, but what I'm saying is that what defines this as threonine is this difference in the R chain. So all of these differences in the R chain lead to what we know as, for example, cysteine, it looks like this, and threonine looks like this. And this difference in the R chain is what makes it unique, and what makes it cysteine is what makes it threonine for this one. So what makes it serine for having this R chain, right? So that's what I really meant, that it's found in the R chain. Like, you're not going to find leucine found in the R chain. Leucine, the diagram was all written out as the amino acid with the difference in the R chain, which makes it leucine. Okay, stereochemistry is the stereochemistry of an alpha carbon is L for all chiral amino acids in eukaryotes. Carbohydrates are D configuration. All chiral amino acids except cysteine have S. Configuration for all amino acids are chiral except for glycine. So these are additional things you have to memorize. So let me just remove this. So the L for all chiral amino acids in eukaryotes, that's the alpha carbon. So it's hard to like understand all of this stereochemistry if you haven't reviewed organic chemistry, literally these terms, because they're all from organic chemistry. So you can think of an amino acid as like a person standing with their arms outstretched. The alpha carbon is like the person's torso, right? So it's a central hub that everything connects to. So it says alpha carbon in little text here, alpha carbon, and it's right here, the central torso, like the center of someone's chest, and then everything is expanding out these 4 compartments, right? And it's a central hub that everything is connected to, right? And all the amino acids, think of it, they have a handedness. So for example, I'm right-handed, you might be left-handed, or you could be right-handed as well. So just like that, you have a left hand and a right hand, and they're mirror images of each other. And amino acids come in left-handed or right-handed versions. So a left-handed version is marked by L, and the right-handed versions are marked by D. So your body only uses the left-handed version. So stereochemistry of all alpha carbons for all chiral amino acids in eukaryotes. Remember biology review that we are made of eukaryotic cells, right? So since we're made up of eukaryotes and all eukaryotes are left-handed, right? That means our bodies only uses left-handed versions of amino acids, right? And then so glycine is a little bit weird though, because like glycine, all chiral amino acids except cysteine have S configuration, and all amino acids except for glycine are chiral. So what it means about glycine is that glycine is the only amino acid that doesn't have any handedness at all. So chirality means you have a handedness, like you can be left-handed or right-handed, but glycine is symmetrical. So if you're symmetrical, you can't be left-handed or right-handed. And that's why it doesn't fit the pattern. And cysteine is another weird one. It's an exception as well, where all amino acids have the same handedness, but cysteine is different because cysteine side chain, or it's R group, remember R group over here, it's a side chain. The reason why is because this R group is different, and so it breaks the pattern. So you don't have to worry about the SR stuff for cysteine, because cysteine is just different. Yeah, cysteine is also weird. It has an S configuration. That's what you have to know. So why should we even care about this configuration thing? What's the importance of knowing, like, oh, this one is a mirror image on the left, it looks left-handed, and the right-handed, blah, blah. It's because your body's proteins are made from left-handed amino acids, and if you accidentally use right-handed amino acids, your proteins wouldn't fold correctly and they wouldn't work. So it's like trying to put on a left shoe on your right foot. So you need, your left shoe with your left foot for the right orientation. So for the MCAT specifically, all you have to really remember is that all the amino acids use this left configuration. and that everything except glycine is not handed at all. Glycine and cysteine are different exceptions. Yeah. All right, and then it also talks about hydrophobic and hydrophilic. So amino acids with long alkyl chains, so a lot of like the carbon chain is super long, are hydrophobic. Those with charges are hydrophilic, and all others fall somewhere in between. So it's important to know. That's why it's important to remember the polarity and also the charges of the amino acids, because it relates to whether they're going to deal with water or not deal with water. In other words, if they're not dealing with water, then they're hydrophobic. If they're dealing with water, then they're hydrophilic. All right, so then we're going to go into actually protein structure. So protein structure is divided into 4. So you can have primary, secondary, tertiary, and quaternary structures. So for primary structures, it's just a linear chain of amino acids in a peptide, stabilized by peptide bonds. And so the amino acid sequence is written from N-terminus to C-terminus. N-terminus is probably charged due to the amino. So I'm going to show a picture. All right, so as you can see the picture, here is the N-terminus, in other words, the N side of the protein for primary structure. And then this is the C side, in other words, this side. All right, so you can see your amino acids here are lined up, right? So this is serine, valine, and serine is there, and so it creates a linear formation. So glycine is connected to serine or beside it, in other words, and then this one's beside valine. So that's why one primary structure is known to be linear, and so what connects them is peptide bonds. Okay, so let's go back to where it said all of that. Why are we on biology all of a sudden? Here we go. So yeah, where do we go? Yeah, sequence chain of amino acids. Right, so we just went over amino acids, and when they're put in a sequence, we get this primary protein structure. Now, for secondary structures, a local structure neighboring amino acids stabilized by hydrogen bonding. So the other one, as I showed, was stabilized by peptide bonds, right? But secondary structure is now stabilized by hydrogen bonds between amino groups and non-adjacent carboxyl groups. And then alpha helices and beta pleated sheets are common in this secondary structure here. So an alpha helix looks like this, and a beta pleated sheet, looks like this. And so that is known for local folding for secondary. And proline is important because it can interrupt the secondary structure because of its rigid cyclic structure. So proline is one of the amino acids that looks like this. Let's see if I can show it. Okay, I'm gonna copy and paste it over proline. Proline here. Let's just paste it here. So nope, that didn't look very good. Paste it here so people can see proline. So proline is one of the amino acids that you're gonna have to memorize as well anyway. And so this one, because of its rigid structure, basically what you have to know is it will start to cause kinks in the structure. A lot of people say the word kinks for that. So that's how it interrupts it. And it kinks meaning it'll start to like form little knots almost, right? So it's disrupting that structure. And then tertiary is over here, and it's a 3D shape. Now it's a 3D shape, right? So we're getting to a 3D shape. We have a single polypeptide chain, so multiple peptides together, and it's stabilized by hydrophobic interactions, acid-base interactions, H bonds, and disulfide bonds. So you have to know what is helping to connect this tertiary protein structure, and it's these bonds here. Hydrophobic interactions means pushing hydrophobic R groups to the interior of the protein, which increases entropy of surrounding water molecules and creates Gibbs free energy. So if you don't understand what any of that means, that's why biochem is like the cherry on top of learning everything, because it's connected to literally everything. It's connected to biology, and it is connected to general chemistry. It's connected to organic chemistry. So really I wanted to do this video kind of last, but someone requested biochem, so I was like, all right, that's fine. But yeah, biochem is like the cherry on top of all of the subjects, really. So hydrophobic interactions is when you push hydrophobic R groups to the interior of the protein. In other words, let's say I have this protein, ball of protein, and basically the part that likes water, or the part that doesn't like water, is on the inside, right? And water is always, like, not always, but there's water in the environment, right? So there's water out here in the environment. And so now the surrounding water molecules, and now the hydrophilic side is on the outside. So that's what you have to know when the protein is folding. So I'll start drawing it on the whiteboard to give like an example. So what happens is that you imagine a bunch of water-hating molecules that are stuck in the water and they don't want to be there. So they don't like the water. Now what happens is these hydrophilic molecules huddle together in the middle of the protein. So this is the protein, right? The region of the protein. And this is in the middle, the hydrophobic part where they don't like water, hydrophobic. Now, like, kids are trying to stay dry by moving in the center of a room during a rainstorm. So these areas don't like water. They want to be dry, right? And so to reduce contact with water, they're no longer touching the water. And what by doing that does is it frees up water molecules that were forced to stay near the hydrophobic parts. They can now move more freely and do whatever they want. So whatever water was here is just moving out freely now, wherever. But it's not going to stay in this hydrophobic region because they don't like to interact with each other. So this makes the system more disordered, right? So now we're getting more disordered or dispersion. I like to think of entropy as kind of like increasing dispersion, or you can think of it as increasing disorder. So there's a bunch of now spreading out and becoming, all the water molecules are becoming very disordered. And so what that does is it creates a favorable energy state delta G. That's where delta G comes in. This is your free energy. And so what this does is this dispersion releases free energy. It releases energy, and this energy release is what drives the hydrophobic parts to cluster together. So these are clustered. Like your body likes the fact of this disorder of the water. So, all of this structure naturally happens. So that's what shapes protein structures. Protein fold themselves so that all the greasy or think of fats, they don't like to associate with water, right? So all the hydrophobic parts are in the middle, inside, and tucked in, right? Away from water. And this is what keeps your proteins together and working properly. Yeah, so that's what you have to know as well. Hydrophobic interactions. And then for disulfide bonds, it's when you have cysteine, which is another amino acid, oxidize and create a covalent bond between their thiol groups, and this forms cysteine disulfide bonds. So if we go and look at a disulfide bond, well, this picture is better. Sorry, I'm getting the picture up here. So if you have these two together, it's a disulfide bond. Di for two, and sulfide, these are two sulfides connected together, so disulfide bond. And when they connect two of these cysteines together, it makes where is it? When you connect two of them together, it makes cysteine. This forms cysteine, whereas this is cysteine as well. Okay, and then for quaternary structure, this interaction between peptides and proteins that contain multiple subunits. So it's basically this multiplied, tertiary structure multiplied. I'm pretty sure a big version that we saw earlier was hemoglobin, HP. Examples. So think of examples of that. And then conjugated proteins and proteins with covalently attached molecules. So covalently means covalent bonding. That's from our bonding section in general chemistry. Covalent bonding. So go back if you don't understand that. Prosthetic group, the attached molecule in a conjugated protein can be a metal, ion, vitamin, lipid, carbohydrate, or nucleic acid. So conjugated proteins, the way you can think of them. Oh yeah, by the way, here's a good diagram of the polypeptide structure. They're showing the C terminus, the N terminus, and then the peptide bonds between them. And these are where they would be. And then these R groups, each of these sections, by the way, this would be an amino acid, this would be an amino acid, this would be an amino acid, this would be an amino acid connected by peptide bonds. The N terminus is this with this amino, and the C terminus is a carbox. The C terminus. Sorry, I just want to go over the conjugated proteins because we kind of got sidetracked with the polypeptide structure. So I wanted to go over conjugated proteins because it actually went over in depth, and I saw that one of the Anki cards was really in depth. So I'm going to go over it better. So you have a complex conjugated protein, and essentially conjugated protein is a complex molecule that combines a protein part with a non-protein part called a prosthetic group. The protein part alone is called an apoprotein, and the combined active is called a holoprotein. So these names and terms are actually pretty important to know, note as well. So if you go over here, we're going to go over conjugated protein. So if we look at common examples, I'm going to use this as well. So let's try to draw what a conjugated protein is. So conjugated protein, oh whoops, it's moving this guy. So conjugated protein, so let's say this is my protein. Now to make it a conjugated protein, I need what? I need a non-protein part called a prosthetic group. I need a non-protein part called a prosthetic group. So I'm going to add the prosthetic group over on this side. Okay, is that a good picture of a conjugated protein? Not quite. But let's just say that this is the prosthetic group for now. Okay, and the prosthetic group can be anything from being like a glycoprotein, meaning it has the protein plus a carbohydrate. So if this prosthetic group was a carb, we would call it a glycoprotein, right? Okay, this doesn't look formatted right. Let me just format it. There we go. You can have this one, and okay, there, that's better. So if you have glycoproteins, it would be the protein plus carb. So if the prosthetic group was a carb, and then lipo would be if it was a lipid. So then you'd have a lipoprotein if this prosthetic group was a lipid, and so on and so forth for the other one. So let's look at an actual picture now of the conjugated proteins. So for the conjugated proteins, if you look at an actual picture of it, why is there no good pictures of conjugated proteins here? So the prosthetic group, the non-protein part of a protein required for the protein to be functional. So the heme molecule of hemoglobin is an example of that. All right. So now we can go over. So this prosthetic group I put on the outside. You probably see it on the inside with the protein, bundled up with the protein structure. So, and then proteins can also denature, meaning the loss of 3D structure. So 3D structure, remember, is tied to tertiary structure and quaternary structure, because quaternary structure is basically tertiary but multiplied. And that's caused by heat or solute concentration. See, it talks about denaturing again here, is when a protein loses its fourth, third, and second structures. So you have to remember that denaturation is a result of losing these, but you never lose the primary structure, right? It doesn't say that explicitly, but you're not losing primary structure. Due to non-covalent interactions such as H-bonds, hydrophobic interactions, and dipole-dipole interactions, those interactions are breaking, the bonds are breaking. And so, in other words, your protein is breaking down. And so, an example, basic example is like an egg, or your hair under a lot of heat, right? Your hair under a lot of heat gets damaged because your hair is made up of proteins. All right. So now I can go over some acid-base chemistry of amino acids. It's basically the same thing that you learn in general chemistry, but applied to amino acids. So I recommend going back to general chemistry if this isn't already clear. Yeah, going back to general chemistry if this isn't already clear, but I'm gonna go over it in terms of what it displays for like amino acids specifically. So amino acids can be a base or an acid, right? That's why they're amphoteric. Water is also amphoteric, but amino acids are also that way. And so they're gonna have a pKa, the pH at which half of the species is deprotonated. So deprotonated as in losing this H plus. And then, where does it say? pH, when it's lowered, the amino acid is fully protonated. When pH is equal to pi, amino acid is a neutral Zwitter ion. So remember that you can be, so Zwitter ion in definition is that you can technically have both a positive and negative charge, and the amino acid structure has that. Remember that we said the amino group is positive and the carboxylic acid is negative. Let me see if I can show Zwitter ion for amino acid structure. Just give me a sec. Well, there's a picture here. It's not that great, but you can see, whoa, why is it never zoom in properly? So you can see the Zwitter ion form. Okay, so the amino group is actually positive, but that actually depends because of pH. So let's say your pH is equal to pi. That's what we're talking about. We have a Zwitter ion where you have a positive and you have a negative. And also it's my fault for saying that. Oh yeah, this is correct. Positive and negative. So I thought I got it wrong. That's actually right. So you have a positive and then you have a negative, right? So the amino group was positive and the carboxylic acid is negative. At a neutral pH, you get an amino acid that has both a positive and negative charge. So it's a Zwitter ion. It can have both positive and negative charges. What a Zwitter ion is. Now, when you have a high pH, you get an anion where only carboxylic acid is negative. And then at a low pH, you get a cation where only the amino group is charged with a positive charge. So review general chemistry if you don't know what a cation and anion is. But general chemistry will help a lot in this instance for learning acid-base chemistry. What happened there? Acid-base chemistry for your amino acids. Okay, so now, yeah, so that we already reviewed how pH is there. So isoelectric point, the pi, so this pi is the pH at which the amino acid is at Zwitter ion form. So it's just the pi at which this amino acid is in that Zwitter ion form that we just saw. So the charges cancel out each other, and that's what makes overall the molecule is neutral. The overall molecule as a Zwitter ion is neutral because positive and negative cancel out. Now, the pKa1 and pKa2 and pKa3, why are there multiple pKas in amino acid? It's because you have different pKas at the different groups so the carboxylic acid group would have a pKa, the amino group would have a pKa of its own, and the side chain would have a pKa of its own. So let's go over the definition for pKa before I explain why there's 3 different pKas for the amino acids. So each group has a pKa, which is the stubbornness of like how easily will that group give up a proton. So if you have a low pKa, the group will easily give up an H+. If you have a high pKa, then the group will easily hold on to H+ tightly. So remember that picture we saw? I don't know if it's on the whiteboard. No, I didn't save it on the whiteboard. That was a good picture. One sec, I'll get it. So let's go over this. So each group can have its own pKa. So this R group can have its own NH and the amino, and the carboxylic acid. Now, they all have a different characteristic for whether they want to really be able to give up a proton. And if they're really stubborn, meaning they have a high pKa, they don't want to give up this proton, right? So at neutral pH, okay, let's explain the values for each of them then. So the carboxyl group has a weak grip, so it's going to let go of the proton easily. So, okay, H+. If I add an H+ to COO, it would have been positive, but right now it's negative. Why? Because look, it has a pKa that is naturally, these pKas are natural, okay? It's naturally having a weak grip of the H+. So it let go of H+, right? So that's what makes it negative. Okay, and then for the amino groups, the exact opposite. It's going to have a medium grip and lets go only at medium pH. And then the side R group varies, right? There's a lot of variation. It just depends. So these all, therefore, have pK 1, 2, and 3, right? Doesn't really, yeah. Now, what the formulas are saying is that it's giving you shortcuts to find the pi. The pH where the amino acid has 0 net charge. So remember that pi was a way to indicate that this amino acid was in its Zwitter ion form, which in other words is its overall neutral form. So even though it's positive here and negative here, overall it's neutral, the entire molecule. So now if we look at the formulas, we go back to those formulas, they're going to tell us how to find the pi, and you have to do a lot of practice questions to get great at that. And then for titrations, I'm going to get a picture for diagram for that because diagrams are important. Okay, so you should be able to see this amino acid titration curve, and it basically follows the same pattern we were talking about. But in titration here, what it says is the midpoint is pH is equal to pKa, and pKa could be, in one of these equivalence point, pH is equal to pi. So equivalence point is pretty important when you do titrations. Now, basically, this chart is not the best chart. This is not a very good chart. I don't like this chart. I don't know if I should use this chart to teach it. Yeah, I don't really like this chart. Alright, so I have a lot of different diagrams to help show what's happening. So in your titrations, right, in a simple titration, this is basically general chemistry review again. I recommend going over Gen Chem acid-base review, but this is the basics. So your half equivalence point is here, where pH is equal to pKa, okay? And then your equivalence point is here, where your moles of your analyte is equal to moles of the titrant. If you don't know what this means, go back to Gen Chem review. I have the video on it already with the chapter is acids and bases chapter. And then this is the endpoint. So now that's just a basic titration, right? And then when you get another titration with two reactions, not just one, so you have one happening here and one happening here. That's all I wanted to show. This is where two reactions are happening, 1 and 2. Okay, now that's one thing I wanted to show. Now with these, you can see you have amino acids where the amino acid has this, can go in this direction or it can go in this direction based on the pH. So when the pH is really high, it's going to go in this direction where the pK is 9.6. And this is for the glycine amino acid. This amino acid here is all glycine. It's glycine that we're talking about. And so the PI is equal to pH at the equivalence point. So PI, so this equivalence point is where PI is equal to pH. So that is where you get your Zwitter ion. So this would be this ion here for glycine, where glycine is neutral, essentially neutral equivalence point. Okay. Yes. That should be the equivalence point, right, guys? And then this blue indicator, if you don't know what that is from acid and base review, which you should know, is your buffer region. Anyway, that's the basics of titration curves, but you have to get good at kind of understanding titration as an experiment and what the curves are showing you. Okay, and then peptide bond formation and hydrolysis. We actually should have went through this after we went through 1, 2, 3, 4 protein structures, but that's okay. So terminology to know is that if you have a dipeptide, you have 2 residues. Tripeptide means you have 3 residues, and oligopeptides have less than 20 residues. So for residues, basically, I just showed this one. So a residue in protein science, a residue is what remains of a free amino acid after it links with others in the protein chain. So basically, when amino acids join, they give up parts of water-making groups. So scientists call these connected units residues instead of full amino acids. All right. So basically, instead of residues, you can think of it as like 2 residues is 2 amino acids connected together. So you can think of it as amino acids. Right, so 2 residues, 2 amino acids connected together, 3 amino acids connected together, and so on and so forth. Polypeptides 20 or greater, right? 20 or more connected together. So forming a peptide bond is a dehydration reaction. The nucleophilic, so this, if you don't know how that works, then you need to go review organic chemistry on dehydration reactions. The nucleophilic amino group of one amino acid attacks the electrophilic carbonyl group. So again, these terms, nucleophilic and electrophilic, are from organic chem. So yeah, go back and review your organic chem for learning about dehydration reactions. But in terms of amino acids, what you have to know is that forming a peptide bond is due to this dehydration reaction. And how that dehydration reaction works will be in your organic chemistry book and review, which I'll be making that video soon. So mi bonds are bonds that look like this: a carbon to attached with bonded to an N, bond of a peptide bond, rigid due to resonance. So it means it's strong. And resonance is something you will, I think it's in the Gen Chem review, and if it's not in your Gen Chem review on resonance, then I don't know why it's not, but it might also be in your organic chem. So basically what I wanted to talk about for CN is that it's rigid, as it was described in the review sheet. So rigid meaning like you don't really like to be, flexible, non-flexible, right? So it's just connected in this one way where it's stuck in this shape kind of thing, right? It's stuck, you want to think of like everything's in 3D formation, not like this 2D shape here, but it's stuck in its 3D formation that it's in. And so it doesn't like to fold randomly or twist, it's very rigid, so it just stays in that way, right? That's what I'm trying to get across for that one. I think that was it. Breaking a peptide bond is a hydrolysis reaction. So you can think of hydrolysis as opposing to the dehydration. So if you want to form it, it's with dehydration reaction. If you want to break the peptide bonds, then it's through a hydrolysis reaction. And remember, the peptide bonds are keeping together this protein for the primary structure here. So now we're going into enzymes. Enzymes are for biochem 2. So if you don't know what a catalyst is, then it would be pretty important to go over general chemistry. But you don't necessarily have to know like enzymes in that light right now, but it would be useful to review Gen Chem on catalysts, because enzymes are basically catalysts, but they're biological catalysts. They're catalysts found in your body, and so the biological catalysts are unchanged by the reactions they catalyze, and they're reusable. So enzymes do not alter the delta. It doesn't alter anything, or it doesn't even alter the final equilibrium position. So this is all things you'll learn in general chemistry. But what they do alter is that they change the rate of the reaction, and that catalyze both the forward and reverse reactions, right? So in exergonic reactions, you release energy. This is now chem review almost. You release energy, delta G is negative. Endergonic, that was exergonic. Endergonic requires energy, delta G is positive. Oxoreductases are enzymes that are working on catalyzing redox reactions that involve the transfer of electrons. So now this is new stuff for specifically the enzymes within isomerases within your body. So these are all biological catalysts. So all of this would be Gen Chem review, except for the fact that enzymes are biological catalysts, and this new term would be enzymes, because enzymes weren't really introduced. You just learned catalyst as a general term in general chemistry. And then so yeah, oxidoreductases are for that. Transferase move a functional group from one molecule to another. Hydrolases catalyze cleavage. Lyses, they're all catalysts, okay? They're helping to do stuff. So catalyze interconversion of isomers. So isomers, and so this is related now to organic chemistry, because our isomers are like different versions of the same molecule, and what the isomerases does is to catalyze that interconversion. So if you don't know what isomers are, or stereoisomers, then that would be your basic organic chemistry review to understand what isomers and stereoisomers do. But isomerases are able to allow that conversion. Ligases join two large biomolecules, often of the same type. Lipases catalyze hydrolysis of fats. So dietary fats are broken down into fatty acids and glycerol or other alcohols. Kinases can add a phosphate group, and it's a type of transferase. So this is a transferase kinases, transferases. And then phosphatases remove, and it's also a type of transferases. And phosphorlases introduce a phosphate group into an organic molecule, notably glucose. So you have to know these different enzymes and what they're doing, and that's exactly what it's written down here. So that's why meltdown is very high yield. So then we have local conditions on enzymes. So local conditions will affect enzymes because enzymes are basically proteins. Structurally, they are proteins, right? So they are proteins, but they function as well as catalysts for many of these functions, as we just saw, depending on which enzyme you are. And so that's why, just like proteins, temperature, pH can affect the protein shape, right? So enzymes can also be affected by temperature and pH and salinity. So temperature and pH can affect an enzyme's activity in vivo. Changes in temperature and pH result in the denaturation. Remember, we learned about denaturation already, of the enzyme, and loss of activity 2, 3, and 4. So remember, we reviewed that up there for denaturation. So it's basically another denaturation review. Salinity in vitro. So let's go over in vivo versus in vitro. So let me just show this. So the main difference in that in vivo tests happen inside a whole living organism, while in vitro tests happen in a controlled lab setting outside of a living organism. So that's what you have to know the difference between in vivo and in vitro. Salinity can impact the action of enzymes. It doesn't really explain what salinity is, so go over that quickly. Salinity is the amount of dissolved salt in the water. So salinity, salt in water. So the salt that's around that enzyme can affect the action of the enzymes. And then Appendix has regulation of enzymes for detailed information on enzyme inhibition. So let's go to that appendix for a more detailed review. And then we'll go back over the terms for, yes, regulation of enzymes. All right, so we're going over finally enzyme inhibition. So I'll put a check mark there. So for enzyme inhibition, what is enzyme inhibition? So enzyme inhibition is basically explaining the process of where a molecule or inhibitor can bind to that enzyme and decrease or stop its activity. So in other words, we have things called inhibitors that can bind to enzymes, bind to it. Like this one is a competitive inhibitor, binds inside of this enzyme, and it inhibits its activity in some way. So we're gonna discover more about that. So Vmax is the maximum rate of the reaction. Remember that enzymes are affecting reactions. They only lower or increase EA, which is activation energy required. They don't change any other thing. Remember that. And then, so the Km is the amount of the substrate needed for the enzyme to work half as fast as it's capable of. So if you increase Km, then it'll lower your enzyme substrate affinity. But if you lower Km, it'll increase your enzyme substrate affinity. So let's go over this. So if I have a competitive inhibition, the Vmax does not change, but your Km goes up. That's what you have to memorize. So in competitive inhibition, your Vmax does not change, but your Km goes up. What does that mean? Then you have lowered enzyme substrate affinity, because the substrate can't even go in. Substrate's blocked because this inhibitor took its place. Substrate's supposed to be able to go in easily. So let's say the substrate's floating around in here, and let's do it green like the other one, so this shape, right? And it's floating around and it wants to go in, but no, it's blocked because the inhibitor took that spot, and that's why we call it competitive inhibition, because they're competing for that spot. And so you have a lowered ES affinity, lowered enzyme substrate affinity, and you can see that these definitions are very helpful while you're studying that. So a competitive inhibitor binds at the active site. So this site where a binding is called the active site, by the way, and thus prevents the substrate from binding. Prevents. No binding allowed. It's that base. All right, and then an uncompetitive inhibition is when the Vmax is lowered, so the maximum rate of reaction goes down. Now, the Km is also lowered because it's an uncompetitive inhibitor, so it's not competing directly for that active site spot. The substrate is still able to bind, but the uncompetitive inhibitor only binds to what we call the ES complex, this enzyme substrate complex, not the active site. And then the non-competitive inhibition is when the Vmax lowers and the Km does not change. And a lot of people might be asking why or how or whatever. You actually don't really need to memorize too much until like that detail. You just have to know non-competitive inhibition, Vmax lowers, Km does not change. Uncompetitive inhibition, Vmax lowers, Km lowers. You don't have to go too in detail. Non-competitive inhibitor binds at the allosteric site. So this is an allosteric site now. Allosteric site. It's basically a different site from the active site. So the active site is over here, active. It does not prevent the substrate from binding to an active site. So the Km does not change. Now, I'm going to explain this in an example. Okay, so let's start with this analogy about cashier at the store. So you have a cashier and you're at a store and you, let's say a grocery store, and there's customers. There's a lineup of customers. So I promise this will help, maybe. So the cashier is the enzyme. Oh, why is everything going away? Oh, 1 second. All right, sorry about that. I was on the wrong layer. So the cashier is the enzyme, okay? And so the enzyme is taking in what we want to call substrate. Now the cashier is taking on the substrate, which is the customers, basically. Customers. So you have cashier and you have customers. And so now the Vmax is the maximum rate at which the max customers that the cashier can say do per hour. Right? So this is the max rate. So the cashier working at a maximum rate, meaning not like their salary, but I'm talking about how fast they go through customers, is their Vmax. So the maximum that they can go through these customers is what is their speed at which they can do it at maximum. That's their Vmax. And then Km is how many customers need to be waiting for the cashiers to work at half speed. So at 50% of Vmax. Right? At 50% of the cashier's Vmax, 50% of their max speed, in other words. Now, how many customers need to be waiting? How many customers will need to be waiting? Let's say it was 3 customers. So Km is 3 in this scenario. So when you have competitive inhibition, you have an inhibitor. So in any type of inhibition, you have an inhibitor, obviously. So I drew the inhibitor there. And now in competitive inhibition, your inhibitor, he's like, or she, is like a fake customer that joins the line and looks like a real customer. So the cashier wastes time on them instead of spending more time with the real customers. So remember that it competes for the active site. And then in uncompetitive inhibition, let's say the inhibitor grabs the cashier and drags them away momentarily to have a chat. So it lowers the rate at which the cashier can do things with the real customers. So they only do this after the customer has stepped up to the register. So this person has already stepped up to the register. They're about to pay, but this inhibitor decides to have a side conversation with the cashier for whatever reason. And so the cashier now becomes slower at their job and slower at getting new customers. So that's why your Vmax not only goes down, but your Km goes down, because you need fewer customers in line now for half the speed because the cashier is already slowed down, right? So to recall what it was in terms of enzymes, the uncompetitive inhibitor will bind only to the enzyme substrate complex. Both Vmax and Km decrease together. Now for non-competitive inhibition, you can think of it as an inhibitor that distracts the cashier by constantly tapping them on the shoulder all the time, tapping them on the shoulder, disrupting the cashier. The cashier now is permanently less efficient. So not only does the Vmax go down, right? The cashier can only do, let's say, 20 customers per hour now instead of 40. So their Vmax is going down, and so they're always inefficient. And so the Km does not change. Instead, it takes the same number of the customers in line to get the half speed. So 50% of their new max, right? So this gets in, non-competitive inhibition gets lowered overall. So it's always lower, like constantly lowered Vmax. So whatever Vmax it was before Vmax initial, now with the Vmax, with the inhibitor final, Vmax final, this new or Vmax new, whatever, with inhibitor, now it's lowered. It's like 20, let's say, right? 20 customers per hour. The other one was 40 customers per hour, so lowered. And so Vmax goes down in non-competitive inhibition, but what happens is that the Km takes on a new number, so it doesn't change anymore. 50% of their new Vmax would be what, right? So that's a basic analogy for Vmax. Enzyme inhibition. And another part of enzyme inhibition is learning that they could be plotted in different ways. So you can have a Michael-Menten curve, or you can have a Line-Weaver-Burg plot. And a Line-Weaver-Burg plot is just a way to show like the linear form of this Michael-Menten curve. So the Michael-Menten curve will actually start with this one, that you at a Km, half your Vmax is here. And what you have to understand is that in the Michael-Menten curve, here's Km, half of Vmax, half of Vmax, half of Vmax, Vmax is here, half of Vmax, Vmax is there, Vmax is over there for the red one, and the black ones is all color-coded to where the Vmax is for competitive inhibitor, uncompetitive, and non-competitive inhibitor. And you have to understand how to read this graph, so when you're shown it, you know that, oh, that looks like, non-competitive, or that looks like uncompetitive. And the important part about Michael-Menten curve is that this is half Vmax. Oh, sorry, not this is half Vmax. The y-axis is your reaction rate, V. V equals reaction rate, as it says here. So all of this is denoting the reaction rate. So now all of this now on your x-axis is denoting the substrate concentration. So if you have a super high substrate concentration for, let's say, non-competitive inhibitor, it wouldn't really go up that much. But if you have high substrate concentration, although it's slow to go up, you will hit your Vmax at a certain substrate concentration for all of these here, right? So you're hitting your Vmax, Vmax, Vmax. And so, yeah. And then for uncompetitive, so you just want to be able to read this and know what's happening. And knowing what the Michelin-Menten X and Y axis is important as well. So V and then substrate. And then line Baker plot is basically the same thing, but except these are line graphs, as you can see. And so it is a double reciprocal plot of basically this. So the reciprocal version, meaning the inverted version. So the slope is equal to Km over Vmax. Some basic math and review. Slope is basically, so let's say I had a line graph and it's going up like this. The slope is the rise over run. Slope is equal to rise over run. So if I want to calculate what the slope is, like if I have a really steep slope, right, I might have a high rise over run. So let's say 10 over 2 is equal to 5. So my slope would be 5, for example. For this one, it's very steep. It's a very steep slope. But this one, let's say the rise is only 4 over 2. Then I only get 2 of a slope of 2, right? So that's slope. And for the slope of this one, it's Km over Vmax. And then for enzymes, this is applied math for enzymes. And then the double reciprocal plot, all of them have the X and Y axes, right? And so your Y axis denotes 1 over V. And it's different. Remember, it's different from Michelin-Menten curve. It's the reciprocal. So the Menten curve has only V, but this is 1 over V for Y. And then for the X-axis, or the horizontal plane, we have this 1 over S, not just S. Okay, and now the left side of the graph is theoretical because you can't have negative substrate or velocity higher than Vmax. This left side of the graph on the left. Vmax and Km can more precisely be calculated using a line break because you're extrapolating out of theoretical values. Michelin-Menten curves show observed values only, not theoretical values. So what it means is like Michelin-Menten had like an experiment, and that's where he gets these curves from. These are all theoretical, meaning you can calculate it out. This makes calculations using Michelin-Menten curve less accurate than Lineweaver-Burke. So this is based on his experiment, and that's what he found, right? So it's less, what does it say here? It's less accurate with calculations. But with calculations, Line-Burke-Weaver plot is theoretical, so you're able to theoretically calculate what it would be. Where would Vmax be, for instance, and stuff like that, with and without an inhibitor. So we're looking at how does this inhibitor change mathematically. So you can do the math of exactly what these inhibitors are doing. And that's why we have these curves to show the rates, or reaction rates, right? Now, Lineberg allows different types of inhibitions to be visualized more clearly. And the reason why is because they are line graphs, they're very straight, whereas the Michelin-Menten curves are curves. So that's about it for all of this part here. Great, we learned about enzymes. Okay, let's go over the terms really quickly. So we already went over feedback inhibition, an enzyme inhibited by high levels of a product from later in the same pathway. Actually, we didn't go over that, but basically enzymes can be inhibited by other things from in a pathway. So as you know, in biochem, there's pathways. So for example, none of the pathways are listed here actually, but for example, you have a pathway where A leads to product B with the help of an enzyme, right? So if A leads to product B with the help of an enzyme, then high levels of a product like B, right? B is made from A somehow. I don't know what's happening, but B is made. And so what B does, well, it would lead an inhibition because of high levels of B is already made. So it's like your body's way of saying, yeah, don't make too much. Like if A, let's add C too for fun, or actually B, let's keep it simple. So A makes B, which is the product. So we have so much of B now, it leads to an inhibition of doing more of this and not making too much of B. So that's a feedback inhibition, it's even feedback. Now, reversible inhibition, the ability to replace the inhibitor with a compound of greater affinity or to remove it using mild laboratory treatment. So what that means is that inhibitors can bind and they can also come out of the binding parts that we were talking about, and that's why it's reversible. Competitive inhibition, we already talked about. Uncompetitive, talked about that. Non-competitive, we talked about that. Mixed inhibition, we did not talk about. When the inhibitor binds with unequal affinity to the enzyme and the enzyme complex, Vmax decreases, Km is increased or decreased depending on the inhibitor has a higher affinity, whether the inhibitor has a higher affinity for the enzyme or enzyme substrate complex. Mixed inhibition. So that's another type of inhibition to know and understand. Irreversible inhibition is the opposite to the reversible inhibition. So an irreversible inhibition is where the enzyme is altered in a way that the active site is permanently unavailable for a prolonged duration, or even permanently. Sorry, it could be permanently or just prolonged, and it kind of disables that enzyme's active site. A suicide inhibitor, a substrate analog that binds irreversibly to the active site via a covalent bond. Cool. So it's basically giving its life away, in other words, committing suicide to be able to basically disable that active site by a covalent bond. So it's pretty much very irreversible. Allosteric effector binds at the allosteric site and induces a change in the conformation. So we did talk about that of an enzyme. So the substrate can no longer bind to the active site, displays cooperativity. So it does not obey Michelin-Menten kinetics. So in positive effectors exert a positive effect, so increase in activity. Negative effectors exert a negative effect, so decrease in activity. So what does it mean to be having a positive effect, increasing activity? That means that if, and this is related to hemoglobin, that if one oxygen binding site binds, for example, it would increase cooperativity. So other binding sites would then be like, oh yeah, let's bind the other sites now. So you have to understand what is cooperativity, how does that work? And then why does it not obey Michael-Menten kinetics? So what it's saying about the Michael-Menten curve is that the Michael-Menten curve doesn't kind of consider that you can have allosteric effectors. What Michael-Menten assumes is that each substrate binds independently and one binding doesn't affect the other. So in cooperativity, we assume that, oh, if a substrate binds to the enzyme, substrate's bound to this enzyme, for instance, then it will affect the other ones nearby. So then this one will be more likely to have activity for a positive effector. If it was a negative effector, then it would be not binding, binding less likely. And so the shape that's made due to cooperativity is a sigmoidal shape, not the hyperbolic curves. So Michael-Menten kinetics has these hyperbolic curves. That's what we call Michael-Menten curves. But for cooperativity, you get something like an S shape, an S, sigmoid. And then homotrophic effector, an allosteric effector that is also a substrate. An example is O2 is homotrophic. An allosteric regulator of hemoglobin, heterotrophic effector, an allosteric regular molecule that is different from the substrate. Phosphorylation is a covalent modification with a phosphate. So catabolism is phosphorylated, means it's active, and anabolism, phosphorylated, means it's inactive. Okay, so for covalent modifications, I want to go over that a little bit. So basically, in phosphorylation, you're adding a phosphate group to an enzyme. So that's what phosphorylation is. And phosphorylation in general is just adding a phosphate group to something. And what that'll do is it changes the shape of the enzyme, and it's able to either turn on or off the enzyme essentially. So in catabolism enzymes, if it becomes phosphorylated, then it becomes more active, right? It becomes more active, increased activity. So it goes faster when you need energy. Anabolism, phosphorylated, inactive, and slows down. So an example would be a light switch. Flipping it on activates enzyme, and flipping it off deactivates it. So your body can quickly turn enzymes on and off without destroying them, and it does it via phosphorylation. So it can make that enzyme inactive or activated by giving it energy or not giving it energy. And phosphate has the ability to do that, right? Glycosylation is covalent modification with carbohydrate. So if you add a carbohydrate sugar to an enzyme via glycosylation, that process, it changes the properties and functions of that enzyme. So now that enzyme, usually it would affect how the enzyme folds, because remember, enzymes are protein. And it can also change where it goes in the cell, or it could change how long that enzyme is going to last. So unlike phosphorylation, glycosylation is usually permanent. That's why you get covalent modification. And the other one that was permanent was suicide inhibitor, right? So permanent. Why am I not using my pen? Permanent. Zymogens are inactive precursor enzymes released into your digestive system as a locked version. So what does that mean? So let's say you have an enzyme released in an inactive form. So inactive form, which is your zymogen. So another enzyme decides to cut it or cleave it. In other words, cleave is probably a better term to use, but if we want to be simple here, another enzyme decides to cut the zymogen, and so that will then activate it. Right, so the zymogen starts off inactivated, but it activates by cleavage. So this is a reaction scheme from Michael-Menten. It's just describing that enzyme substrate can lead to enzyme plus product, or enzyme plus substrate is the ES complex here. So this is related to enzyme kinetics. Yeah. So to simplify things, we can also assume none of the product reverts back to ES, which is true at the start of the reaction. This is why K minus C was omitted from the reaction scheme below. Okay, enzyme kinetics is where you get saturation kinetics, where if you increase substrate or in the environment, so you have your enzyme here, and there's substrate all around in the environment, and if I increase the amount that it's there, it would increase the reaction rate until a max value is reached. So remember Vmax, right? So the graphical plots, we already went over that. Weaver-Burke is a line plot, and Michael-Menten uses curves. I keep saying Mentes. Km we went over, and Vmax we went over, but we didn't really go over this specific formula, so this formula would be useful in practice problems. And then Michael-Menten equation would also be useful in practice problems, which I'll show you soon on how to open those up. And then cooperative enzymes display sigmoidal curve. So we already showed that over here when we were talking about allosteric effectors and cooperation. It's a sigmoidal curve, as I drew. Whoops, now I deleted it. As I drew over here, it's like an S shape of a curve on the graph because of the change in activity with substrate binding. Mechanisms of enzyme activity. Enzymes act by stabilizing the transition state, providing a favorable microenvironment or bonding. So anyway, you just have to know that it's stabilizing the transition state, and that's why enzymes are helping with the reaction. So the active site is the site of catalyst. We already talked about active site. Now we're going into theories about the binding between the enzyme and the substrate. So substrate. Substrate. Let's say this is your substrate. And that's an inhibitor. Let's remove the inhibitor. We're not talking about inhibitors anymore. We're talking about binding into their enzymes binding with their substrate. So there's a lock and key theory where the enzyme and substrate are exactly complementary and fit together like a key and a lock. Simple enough, right? And then there's induced fit theory where maybe the enzyme isn't made for the substrate right away. Instead, when the enzyme and substrate kind of interact together, they undergo conformational changes in order to interact fully. So let's say this was more like rounded edges and this was rigid edges. So then when they come together, they're both changing like shape. The ES complex over here is trying to change a little bit to be able to fit perfectly. And I believe this is the theory that's favored today in biochem. Cofactors is when you have a metal cation that is required, so this would be something you review in chemistry, what this term means, that is required by some enzymes. And coenzymes are organic molecules that is required by some enzymes. So some enzymes have special requirements that could be cofactors and coenzymes. Now we can get into non-enzymatic protein function and protein analysis. So in protein isolation, there's more detailed information in the appendix about lab techniques. Lab techniques are really important to learn. Don't ignore your lab techniques. Let's see if we have enough time to go over those as well. We should be able to go over everything pretty in detail. I'm just going to get water. Then for biochemistry, we have non-enzymatic protein function and protein analysis, so cellular functions of proteins. So now we're going into more like different types of proteins, but more specifically what function do they have for the cell. So structural proteins compose the cytoskeleton, anchoring proteins, and much of the extracellular matrix. You should understand what these terms are. The most common structural proteins are collagen, elastin, keratin, actin, and tubulin. So if you remember biology, that would be important because you would understand where these are coming from. They are generally fibrous in nature. Motor proteins have one or more heads capable of force generation through a conformational change. So remember we were talking about in biology, so I would advise to review biology before you learn cellular functions, because in biology we're reviewing the muscular system, right? And part of the muscular system is understanding myosin, kinesin, and diasin, and those are examples of the motor proteins. So they have catalytic activity acting as ATPases to power movement. Common applications include muscle contraction, and we learned that in biology, vesicle movement within cells, and cell motility. So there's other different ones other than just muscular contraction. There's also vesicle movement, yes, and cell motility. You also have binding proteins that can bind a specific substrate either to sequester it in the body or hold its concentration at steady state. You have also CAM molecules, CAM, which stands for cell adhesion molecules, and they allow cells to bind together to other cellular surfaces. So they're kind of like structure, they're adding more structure, they're holding things together. So cohedrins, integrins, and selectins are types of CAMs. They're underneath the umbrella of CAM. So cohedrins are calcium-dependent glycoproteins. So remember we talked about proteins that can be conjugated. A glycoprotein, so remember over here, we're talking about how you can have conjugated proteins with a prosthetic group. And one of our examples on the whiteboard was glycoprotein. Now it's coming back over here. So where is it? Calcium-dependent glycoprotein that hold similar cells together. Integrins have two membrane spanning chains and permit cells to adhere to proteins in the extracellular matrix. Selectins allow cells to adhere to carbohydrates on the surface of other cells and most commonly used in the immune system. So going over biology of the immune system and all of biology would really help for understanding, connecting it back to cellular functions. Antibodies, that's connected to our biology unit on the immune system. So going over back to that biology video, you would understand antibodies, also known as immunoglobulins or IG, are used by immune system to target a specific antigen. We already went over antigen in bio, so in the bio video, so I won't go over that again. But antigen's like that harmful thing that is detected, right? Which may be a protein on the surface of a pathogen or toxin. The variable region is responsible for antigen binding. So those are different proteins in your body and their function. So antibodies, again, are proteins, and this is their function. And this should be reviewed from bio. But the new light that it's shedding over here in biochemistry is that antibodies are specifically proteins, and they're a type of protein that have this function. Okay, let's go over, before we go over protein analysis and protein isolation, which are basically like lab-like type of things, we're going to go over biosignaling. So how do things talk to each other? How does your cell know what it's going to do and cascade in effect? So ion channels is a big way that this happens. This is related to action potentials. Remember in biology, we were talking about action potentials and how your cells like to communicate. Like a neuron will synapse with another neuron, or even an effector. So remember our postganglionic and pre and postganglionic neurons can talk to each other. And so if this one releases its vesicles and, has its vesicle here and releases its neurotransmitters to the other one with a receptor, so it's waiting with the receptor on the other one, right? What happens is that can cause then channels to change. So ungated channels are always open, but voltage-gated channels open a range of membrane within a range of membrane potentials. So let's say that this neuron changed the membrane potential of the next one, or not change the membrane potential, but let's say you had an effector with a membrane, like muscle, right? That's another way it can communicate, right? And so if the electrical signal depolarizes this membrane of whatever's coming next from the one before it, this depolarization, okay, we're talking about action potentials, by the way, so if you're confused at what the heck's happening, we're talking about action potentials. And so if you depolarize that, then it will then cause voltage-gated channels to potentially open if it's depolarized enough. So if it was within a range of, let's say, plus 15 millivolts to whatever, 20 millivolts, I don't know, or something like that. It's just a change from resting membrane potential to a different, so the voltage-gated channels will open due to that electrical signal, right? And then ligand-gated channels open in the presence of a specific binding substance, usually a hormone or neurotransmitter. So I already reviewed that basically, so check mark. Enzyme-linked receptors participate in cell signaling through extracellular ligand binding and initiation of second messenger cascades. G-coupled protein receptors are basically other types of receptors as well, but GPCRs, we actually went over this in biology, so the biology video is helpful to review, has a membrane-bound protein called G protein, which has alpha, beta, gamma subunits. So this G protein has those units in them, and the first messenger ligand initiates the second, and it creates a cascade response. So this is like an example of a cascade response. So the first messenger is epinephrine, and that is the ligand. At the end of the GCPR process, the phosphodiesterase deactivates cyclic AMP, and GTPs hydrolyze the GDP. So it creates this response later on, and CAMP is the secondary messenger, and then it's able to, this hormone then can cause these effects on the body because this cascade has happened successfully. Okay, now we can go into protein isolation. There's more detailed information in the appendix for lab techniques, so I recommend going over lab techniques. We might go over this after that, so we will go into the details afterward. So electrophoresis, because I want to give a broad overview of the different techniques before we go into details. So electrophoresis, when you use a gel matrix to observe the migration of proteins in responses to electrical field. So let's say you have an electrical field with plus side and a minus side, and you have gel in here. And what you basically do is you put proteins in that gel, and you see where do those proteins move. In other words, their migration. Okay. And then native page maintains the protein shape, but results are difficult to compare because the mass and charge ratio differs for each protein. An SDS page denatures the proteins and masks the native charge, so the comparison of size is more accurate. So size is more accurate, but functional protein cannot be recaptured from the gel. Isoelectric focusing is when you separate proteins by their isoelectric point. So remember we reviewed PI. PI is where the amino acid is neutral and it's a Zwitter ion. Remember that, PI? And so the protein migrates toward an electrode until it reaches a region where the gel pH is equal to the PI. So then you would also figure out what pH would lead to the Zwitter ion being, neutral. Would lead to PI, which means that you have a Zwitter ion, which is neutral. I will go over these two in more detail soon. It's probably in the appendix, so that's why I didn't want to expand too much yet. Chromatography is when it separates protein mixtures on the basis of their affinity for stationary or mobile phase. And then you have different types of chromatography underneath here. So the different types of chromatography include column, ion exchange, size exclusion, and affinity. So I'm going to go over all of these in more detail because lab techniques are actually very important, as I said earlier, to go over. So let's go over those. So reading out all of this text is probably not going to help the average viewer or the average person, just because it's very hard to visualize these lab techniques, because you want to think of it as you are a scientist. You're a scientist who has entered a lab. And reading this is like, nobody reads the okay, some people and most people will be able to read the manual and understand, how to build the thing that they're building, or the procedures. You get procedures from the manual, an instruction manual of, installing a fridge. But what's going to help more is pictures. So every manual has pictures. This manual does not have many pictures. It has one for gel electrophoresis, and that's it. So we're going to teach this in terms of pictures and diagrams instead. So all of this, I'm just going to do on the whiteboard too now. Alright, so we're going to start off with electrophoresis. So on the right here, you see the mile down, so we're following that pretty carefully, but this time I'm just going to draw things out. So I'll just flip the device over. So gel or electrophoresis. So we start off with a gel. Think about a gel like shaving, not shaving gel, what is it called again? When you do your hair gel, kind of like that, I guess. You start, not with hair gel specifically, but a gel. And so what I'm explaining right now is just gel electrophoresis, the general setup. And then we'll go over the different types that's happening over here. So the general setup is you have the gel and you have negative particles at the top. Let's say you have negative here, negatively charged. And then positive, I'm going to do in green. Oh, didn't change the color, green. You have here at the bottom. Now, arrows showing the particles moving downward, because why? Why do we want the arrows from negative to positive? So imagine you're in a crowded hallway filled with jello. So you are the negative side over here, and you're traveling to the positive side to be, you want to join the heaven. I don't know. And so you drop a bunch of tiny charged balls at the top. And let's say I turn on a magnet at the bottom that pulls the charges downward. What happens? So right now, let's say that this is not on right now. Like, this is not on. So nothing's moving. But when I turn this on, right, the negative goes to the positive, because in this case, opposites attract. So then what happens? The ball races downward. So that's what I wanted to explain. But here's the thing. Bigger balls get stuck in the jello and move slower. So you're dropping these balls, and the bigger ones are getting stuck in this gel area here, this middle area, because they're too big to pass through the gel. And the smaller the gel, the smaller molecules able to go through and fit easily and travel to the positive side more easily. So this is how we can separate molecules by their size. So this is what it says. Separates macromolecules like proteins, DNA, RNA. For proteins and small molecules, the gel is, so the specific gel name, by the way, is polyacrylamide, as it says here. For proteins and small molecules. For large molecules, the gel is agarose. Give me 1 second. Alright, so there's different types of gels, as we just explained. Another thing to note is that, so that you can also stain these balls or particles to be blue, so that you can actually see what's going through and what's not, kind of thing. So stain for visualization is what it means there. Now for the different types, if you have a native page, it's using a polyacrylamide gel. So with polyacrylamide gels, remember, or acrylamide gels, is that it's for proteins and small molecules only. It's a method for proteins using non-denaturating conditions. Proteins keep their native charge and structure, so they are separated based on charge and size. So remember that native page will be able to separate molecules by charge and size. And then for proteins, so you're testing proteins based on their charge and size. And the SDS page is still the same gel, for proteins, right? Because that gel is used for proteins, using denaturating conditions. So this sodium dodecyl sulfate, I don't know how to say it that well, denatures the proteins. Remember we talked about denaturation already, gives the proteins a uniform charge. So what SDS does is that, remember, native page will be able to test, oh, the protein is a certain charge and a certain size, right? So that's what it can test. But what's happening in SDS page, as SDS denotes, is you put SDS, the sodium decyl sulfate, onto all the protein particles, whatever, and what will happen is all of them become the same charge. So all of them will become, let's say, positive, or all of them will become, let's say, negative. So what it's trying to do, is give it a uniform charge, and this allows them to only be separated solely on mass rather than mass and charge. So that's the difference. Thus, you can estimate the protein's molecular mass only, so the size only, because we already controlled for the charge, with SDS. So that's the SDS page. Now, the reducing SDS page is the exact same as the SDS page, but with the addition of a reducing agent. So you have this beta, whatever, mercaptoethanol, which will reduce the disulfide bridges and result in a completely denatured protein. In isoelectrical focusing is when the gel electrophoresis method separates the proteins on the basis of their relative contents of acidic and basic residues. The gel has a pH gradient and proteins will migrate through the gel. So this gel instead, for isoelectric focusing, has a pH gradient in this gel, right? And so proteins will migrate through the gel until they reach the pH that matches their isoelectric bond. So remember, Pi will give you this Witter ion. pH will, yeah. And it'll give you the pH of where that protein is at the Zwitter ion, or the neutral charge, basically. Remember, Zwitter ions were neutral charges. I had to reiterate the main concepts again and again because those are important. So it'll no longer be attracted to the anode, and it'll stop migrating. So it'll just stay in this, within the gel, wherever the pH is more leading it to be neutral. Because it's basically no longer charged, and if it's no longer charged, then it just stays in its place because it's happy being neutral. And also because it's like not negative anymore, so it's not going to be going to the positive side. If it's neutral. So southern blotting is a detection of specific DNA sequence in a sample. So this is for detecting DNA sequences. And then northern blotting is detection of specific RNA sequence in a sample. And western blotting is detection of protein in a sample. So let me go to picture. So here's a Google image picture. And it's pretty good. So it's using, again, agarose gel for large, okay, this one didn't. These two are using it for large molecules, and this one for smaller, right? And so you have a cell with DNA, and your cell has all this different stuff. It has DNA, RNA, and protein. So if you want to test for this, you could use gel electrophoresis, like we just said. But what blotting is be able to then print your results out. So I want to then print my results out, kind of thing. So I found my blot. I did electrophoresis, and now I'm going to put it on a southern blot. So I did my electrophoresis from this experiment here, and now I'm going to put on a southern blot, which will be able to give me the DNA sequence of the sample. And on the test, it's actually even more specific than that, because you can, basically, they're going to give you, like, the sequence looking thing, and then it's going to be like, what does it show you about this, right? And so you're going to have to be able to do that as well, which will get better practice through a good practice question bank. A lot of people use UWorld, for instance. And then the northern blot, is exactly that. So you have the northern blot, and it's done the same thing. You've done the experiment, and now you can put the sequence onto a blot, which will be able, you could read the results of exactly what the northern blot shows you, which is a specific RNA sequence. So it's for RNA. And then for proteins, you can put them onto a western blot with this, you're using this gel, by the way, and you can, for electrophoresis, and you can put it on your western blot and be able to figure out, you know, the specific protein that was in the sample. Yeah, and then so the results will give you something that looks like this, and on the test, you basically want to be able to learn to read these. Okay. Alright, so we're going to go over how to read a blot. So because this is generated by an AI, not everything is going to be 100% accurate. I'm going to go over exactly the basics idea of what you should actually take from it. So in this bottom part, it says same 3 steps every time. So the first step is separate by size on a gel, or in other words, to do your gel electrophoresis experiment. Gel electrophoresis. And then when I told you guys that basically blotting is printing out the results, that's what you're going to do. They take what's happening with gel electrophoresis and they transfer onto a blot, or they transfer the blot onto a membrane. So these are membranes that you're seeing here. So they're able to transfer the results onto something that you can see. And then the last step is the probe with something that finds your target that you're looking for. So let's go over how to read the blots. So the first one we have here is the Southern blot, which detects DNA, now the DNA sequence specifically. And it's labeled with a DNA probe. That's why you need some sort of probe that finds your target. So in this case, our target's DNA, so we use a DNA probe, simple enough. And each of these, you notice, has a ladder. As you can see, it wasn't generated very well, but basically this column here, sorry, I don't know why I drew a line like that, but this column here, the gray column, is basically the control, where you compare the other conditions to. So for example, sorry, this is the control, but the ladder is basically able to tell you, able to be like a comparison, right? So you're comparing your results to that ladder. So what we're going to do again is how to read the blots, but before anything else, we want to understand what is blotting in the first place. So imagine you have a messy soup of molecules, right, all mixed together, and you want to know, is this my specific molecule of interest, which could be, I don't know if I can draw on this, but it could be DNA, RNA, or protein. Blotting is the lab technique that answers that. So we already went over that gel electrophoresis can sort by size. Also note that it could also sort by charges depending on the type of electrophoresis you have, and then you're going to transfer that and then probe. So we already know that. And then the analogy for this, or analogies for this, I don't want to go over analogy because we already went through this part thoroughly. So now let's say you have a Southern blot which is able to detect DNA sequences. Now, so the suspects in the lineup are DNA fragments. So these are DNA fragments, as you can see here. The wanted poster is a labeled piece of DNA that only sticks to its exact matching sequence. So let's go over how to actually read this step by step then. So step one is to find the ladder first. Here's your ladder. And every blot, as you notice, has a ladder. So even Northern blot has a ladder, and Western blot also has a ladder. Now, this is like your ruler. Each rung has a known size. So this has a known size. So nothing else means anything until you've all lined these up. So these are always compared relative to the ladder. So if this is at 2 kb, then we would know, oh, these are also at 2 kb. I mean, sorry, these ones in this row are also at 2 kb. Position is equal to size. So if you're higher on the gel, it means you're bigger fragment, so then you moved less, right? So if you're higher up, you're larger. You're larger your size is larger, right? So it moved less. But things that are smaller, small particles, can move further down the gel, right? So that's why we see them more at the bottom here. So this one is smaller and it's closer to the bottom. Now our step 3 is to compare every lane back to the control. So remember, that's what we were just doing. So all of these we can compare to the control, which is 2 kb. We can compare this one, which should be 1 kb. Okay, so patient A's band sits at the same height as control. So if you look at patient A, they have this one at the same height, right? And patient B has a second lower band not seen in control. Most often a deletion made that fragment shorter. So you can see something has changed here, and this band is lower than this band. So then step 5 is 2 bands in one lane, so heterozygous. So as you can see, you can have 2 bands happen in one lane, and patient B kept their normal size and gained a shifted one. So this is their shifted one. They carry the normal one in the mutant copy. So that's an example of a Southern blot, a general idea of reading one. It might not show up in this way, actually, to be honest, because there's other DNA testing that this is mostly just an example. It's not, exact of what you might see in different problems. In the Northern blot is the same lineup trick, but the suspects are RNA transcripts, and the poster is a probe that hybridizes to one specific mRNA. So you're trying to detect mRNA. So it's the same ladder logic as last time, so we don't have to go over that again. And what we're looking at is the presence or absence. So is the gene on? So a band means that tissue is transcribing this gene right now, and no band means it's not. Right? So in this band, strong dark band, this gene is highly expressed here and lots of mRNA copies at this point, at this size. Same band, but faint. The gene is expressed here too, just at a lower level, and then here is none, right? I don't know if you can select it there. Whoops, why is it? It's hard to select here. Brain, no band at all. This gene is not being transcribed here. So the darkness is, by the way, this is all very oversimplified ways to read it. When you get to practice problems, like a U-World Q-Bank, for example, is going to be harder. Darkness is how much. So a darker, thicker band means more mRNAs are present. So this is dark and thick, right? And the presence and absence, by the way, a band means that the tissue is transcribing this gene right now. So let's say there was no transcribing. This would be just absent. There wouldn't be like a dotted line like this. It just wouldn't show up, by the way. And then for step 4, the same height across lanes, same transcript, all bands sit at the same size here. So as you can see, all these bands sit at these two sit at the same size. If one lane's band were shifted, this one is technically not there. Just pretend it's like not there, invisible. We just had this here for reference. If one lane's bands were shifted, that would suggest a different splice variant, not just a different amount. So if you have a different height, then it would suggest a different splice variant. And now we're going to look at the Western blot, which detects protein. So it's very similar. We have the ladder. And so step 1, we're going to look at the position of the ladder here, and we're going to compare it relative to the ladder. So the results here showed that, okay, what we're measuring on the ladder is no longer kb. Like these ones were both kb. But notice for Western blot, it's going to be kDa on this side. So that's how you know the antibody found the right thing. So this one, what we're doing here, is we're looking for a particular protein. And one way you can find it is like in electrophoresis, so you can use like some sort of antibody or some sort of thing and realize that, oh, this binds with this, and so something is happening. But yeah, that's you gotta do basically the experimental technique and then do these results to get that. And so now the darkness is equal to the abundance. So since this is dark, right, it's the same size as these ones and these ones, but the difference is that even though it's the same protein, there's much more of it after treatment. So same loading control band, same intensity as other. So in untreated protein, there's less, but it's the same over here. So what is the knockout lane in our specific results here? So this is where no band, where the protein generally can exist, confirms the antibody isn't binding junk. And step 4 is never trust the target band alone. Always check the loading control first. So the loading control, equal loading control confirms all 3 lanes had the same total protein amount. So the target band differences above are real. So loading control is basically in the name. What you're doing with the loading control is that when you run your electrophoresis gel thing, right, that experiment, you need to confirm that you loaded equal amounts of the total protein in each lane. And it matters because imagine you're comparing a target protein across 3 different samples. If lane A's band is darker than lane B's, or if lane A is lighter than lane B, for example, you might think that, oh, there's more protein in lane A just because it's darker. But what if you accidentally loaded twice as much total protein in lane A? Then, of course, the target protein band looks darker. It's not that A has more of that specific protein, you just loaded more stuff overall. So what the loading control fixes is this. You pick a protein that should be present in roughly equal amounts everywhere, and you detect it on the same blot, usually at the bottom of the gel. And if all 3 lanes show equal intensity loading control bands, you know you loaded equal total protein, right? And any differences in your target, so that's a control that you want to do. And any differences in your target band above it are real biological differences and not just differences in how you loaded it. So that's why it has to be kind of like a control variable. Okay, so let's just do a review. So Salem blot finds DNA, and we use a DNA probe. The fragment size, mutation if shifted, and the band intensity tells you mainly presence and the size. And then for Northern blot, it will find RNA. The probe will use RNA probe, and transcript size, the splice variant if shifted, and it will tell you the expression level, so how much mRNA there is. Western blot will show you the protein, usually using antibody, right? And then molecular weight confirms the identity. So, and the band intensity itself will tell you the protein abundance. So the band position and the band intensity will tell you something, and that's how you read a little bit, like a very basic of your reading the blots. All right, going back to the meltdown, we went over this, this native page, SDS, reducing, isoelectric, southern, northern, western, in a lot of detail. So now we're gonna go over, so big check mark here, and now we're gonna go over chromatography, which is when you can separate 2 or more molecules from a mixture, and it's almost like filtering. Let's see if I can write this out, filtering. It's kind of like filtering. Like if you ever filtered coffee and stuff using the coffee filter. Yeah, it's kind of like that. So let's see a diagram. So as you can see here, let's see if we can use a Khan Academy. They have principles of a good article here too to look at. So you can go on Khan Academy if you're still confused about chromatography and see that basically you're going to have, I don't know why there's so much here going on. So basically you're going to have something that looks like this. And you basically have a filter kind of thing here. And so your eluent, this one, is a fluid entering, any fluid entering the column. And then you have mobile phase and stationary phase. So your mobile phase is the solvent moving through the column. If you don't know what solvent and solutes are, you need to review a chemistry solution unit on solutions. And then stationary phase is substance that is fixed inside of the column. So if we go back here, stationary phase is typically polar, and polar molecules elute slower. So they're going to go down that thing slower. And now the mobile phase is typically nonpolar, and they're going to go down faster. And so the liquid chromatography is when you use silica as the stationary phase, while toluene or another nonpolar liquid as the mobile phase. So I'll go over these from I guess an interactive viewpoint in a second. High performance liquid chromatography is a type of liquid chromatography that uses pressure to pass the solvent phase through a more finely ground stationary phase, which increases the interactions between, this is a typo, molecules and the stationary phase. HPLC, the high performance liquid chromatography, has higher resolving power. So we'll go over that soon as well. Now there's also gas chromatography. Gel filtration chromatography is also known as size exclusion chromatography, ion exchange chromatography, affinity chromatography, and thin layer chromatography. So for the size exclusion chromatography, it separates molecules by their size rather than the polarity. So larger molecules will elute faster, they'll go down faster, right? But the smaller ones won't. Yeah, and then ion exchange chromatography separates proteins by their net charge. So cation exchange is when negative beads are used so that the negative proteins elute first, and then anion positive beads are used so that the positive proteins will elute first. In affinity chromatography, it's about binding, seeing how beads are bound to specific ligand and proteins with high affinity for that ligand. So proteins with high affinity for a certain ligand will bind, right, because they want to be together. So the proteins with low affinity will elute first. For thin chromatography, there's a polar silica gel, and molecules are spotted on the bottom of the sheet. Sheet is placed in a nonpolar liquid, so the mobile phase travels up the plate using capillary action. Nonpolar molecules have the highest RF value. So we can go over the basics of chromatography because, again, lab techniques are important to understand. So chromatography is a way to separate a mixture of molecules based on their different properties. Like if you have a complex soup of stuff and you want to pull out a specific thing, it's basically what I said before, it's filtering. Now, the key players is the stationary phase and the mobile phase. Every chromatography technique, all types of chromatography will have a stationary phase and will have a mobile phase. So the stationary phase stays put and it doesn't move. That's why it's called stationary. And mobile phase, you can guess that it's flowing and moving, in other words. So usually a stationary phase can be a gel, beads, or a packed column. And this is the place or the track that the molecules will interact with. Molecules that like the stationary phase will stick to it longer and travel very slowly. They'll travel very slowly through this column. Okay, so mobile phase is like the river carrying the molecules along. So it's helping to carry this along. Try to ignore that this text here that's overlapping with the column. And so the core principle is that the molecules are separated based on how much do they like the stationary phase versus how much do they like the mobile phase. And this is the really basic idea of chromatography. So in liquid chromatography, you have your stationary phase, which is silica gel, that's polar. And then the mobile phase is toluene or another nonpolar liquid. So what you're doing in here is that like attracts like, so the polar molecules will be attracted to a polar silica and then stick longer, but non-polar molecules won't like it and they'll rush past. So based on whatever elutes first, down this column will tell you whether it's more nonpolar or polar. And then technique 2 is HPLC. It's the same idea for liquid, but it has a high pressure and a finely ground stationary phase. Okay, and then gas chromatography is when your stationary phase is a thin layer on the inside of the column, usually polar, but the mobile phase is a gas like helium or something like that. And so what it's going to do is separate the molecules based on two things, polarity and its boiling point. So anything with lower boiling point that's really gas-like will elute faster. And polarity is just like in any regular liquid chromatography that we just went over. And in gel filtration chromatography, size exclusion, the porous gel beads like tiny sponges with holes. So the stationary phase is basically like a sponge with holes in them, and it's trying to see, like, we're trying to filter by size. That's why it's also called size exclusion. So the pores are key, and imagine a bead with holes in it. So large molecules are too big to fit inside, so the small molecules can squeeze through, and then they'll come out the end of the column. So that means you separate the molecules purely by size and not by polarity or charge. So the biggest molecules will elute first and the smallest ones will elute last. And there's a good analogy here that you can think of it like runners trying to get through the crowded street. Tall people or big molecules can't fit between cars and then they have to run around them. So they're going slower. They move fast. They have to go around them. They move fast and get to the finish line first. But small molecules can weave between the cars and they take longer. So this picture is not that good, by the way, but this explanation is good where it says that large molecules come out first. And why do small molecules come out last? Is because imagine they're getting stuck and they're wandering through a maze, right? So they have to go through that maze to get out of the column, so they're slower to travel than the large molecules. And then lastly, or second last, sorry, or third lastly, is the ion exchange chromatography. So your stationary phase is charge bead and mobile is a buffer solution. So basically the rule is that opposites will attract. So if the bead is negative, then positive proteins will stick longer. If the bead is positive, then negative proteins stick the longest. So proteins and other molecules always have a net charge, and they're either overall positive or negative, and the stationary phase beads are also charged. So we're trying to see, based on opposite charges, will attract. And then affinity chromatography is also a mobile phase that's a buffer solution, and you're using beads. And you're looking for one specific protein. Your target protein finds its ligand and the beads on the beads and binds tightly. So affinity is about binding, and everything else doesn't recognize the ligand, so it passes through. So it's like a lock and key theory or mechanism. So everything else that doesn't match will flow through the column fast. And then thin layer TLC, a polar silica gel coated on a glass or plastic sheet, and the mobile phase is a non-polar liquid. And so you basically it's a very fast, cheap, and easy way to be able to first spot your mixture at the bottom of the sheet, and then you usually dip the bottom of the sheet in a non-polar solvent so that the solvent will crawl up the sheet. So then your polar molecules will stick to the polar silica and they won't travel far. They'll just stick at the bottom. The polar silica is over here. So they're sticking to the more polar side because polar stuff wants to be with other polar stuff, right? And the non-polar molecules love the non-polar solvent. So the non-polar solvent is over here, I guess. I don't know, this diagram's not that great. I think that there's a better one on Mile Down itself. So if we go there, see here's the solvent front. So this solvent front itself is not polar or non-polar. It's just the physical line that the liquid mobile phase reaches on a chromatography paper or plate. The actual liquid, the solvent, or the mobile plate, like the liquid, is creating that front and can be either polar or non-polar depending on your experiment. So the liquid is the solvent, and polar or non-polar, depending on what you want to experiment with. And so non-polar molecules have the highest RF retention factor. So what retention factor basically is that if you have non-polar molecules, they have a high RF because basically, how do I explain this? Let's say your plate overall is just, this plate that you have is overall polar, right? So we chose that it's polar in this one. And so the molecule is nonpolar, so this nonpolar one, so it doesn't like to stick to the polar plate, so it stays completely free from it, and it's able to travel. So the solvent carries it, and the moving liquid easily sweeps the nonpolar molecule right up to the top, where the solvent front is. So because the nonpolar molecule travels almost as far as the liquid, it gets a high retention factor, which is close to 1. So the retention factor. So the retention factor is your distance of the spot moved over the distance solvent front moved. And basically, it would be closer to 1 because it's moving almost as fast as the liquid, or almost as far as the liquid. All right, and then we'll quickly go over DNA sequencing. So you can think of DNA sequencing as you're trying to figure out the exact letter sequence of DNA. So remember that review of, I think, Biology 12, or actually there's a DNA sequence here to show you. So you have a DNA sequence, for example, and you can think of a copying machine that jams randomly. So you're going to mix your DNA template with normal nucleotides. So the nucleotides, that's how you get a sequence like that. Your nucleotides are A, T, C, G, right? So I think it's adenine, thymine, no, not that one. Nucleotide and guanine. C would be, where did I put this? C would be over here. Types. I just want to show different types that you can have. No, here, like that. Cytosine is there as well. Cytosine, thymine, guanine, adenine. So they're making up these DNA sequences. And so if you have these plus a small amount of poison, we're just gonna call them like an analogy, like it's poison. It's not actually poison, but let's say you add this plus DDNTP, DDNTP here. So what's gonna happen is that you have those added with the normal nucleotides. Now, each missing the 3' hydroxyl group that DNA polymerase needs to attach the next letter. So whenever a DDNTP gets used instead of a normal nucleotide, so let's say a DDNTP is used in a sequence like this instead of a normal nucleotide like thymine, for example, then what happens is that the chain just stops there permanently. So the chain ends with DDNTP, which is marked with a colored dye that it blah blah blah, right? And so since this happens randomly across millions of copies, you end up with a huge mix of fragments of every possible length, each one ending in a DDNTP that's color-coded by its base. So you would basically have different dye colors for A, T, and C, and G, right? And so you run that mix through a gel electrophoresis, which sorts the fragments by size. So the strands run through gel electrophoresis sort based on length, which is basically size. And so it matters because this is a method to be able for scientists to read DNA before the next generations of sequencing takes over. It's slower, but it's the gold standard for short, precise reads of what a DNA sequence would be. So that's why it's called Sanger DNA sequencing. And then your polymerase chain reaction is when you're trying to make millions of copies of one specific stretch of DNA. So specific DNA region and copies of it. So let's say you want to copy DNA, right? So what do? You want a DNA photocopier. And that's what polymerase chain reaction is, what we're trying to do. So with 3 repeating steps. So here are the steps, 1, 2, and 3. So the first one is denaturation, where you need to melt the DNA double helix into 2 single strands. It's like kind of like you're trying to pull apart the protein like a zipper or something. And then annealing stage, and so you need this temperature for it to happen, right? For it to happen. And then so annealing is when you cool it down so that short DNA snippets called primers can stick, and that's what we call annealing to the start and end region of where you want it to be copied. So primers need a high G and C content, and G and C right at each end, because GC pairs bind tighter. They have 3 hydrogen bonds. So these, I'm talking about the nucleotides, G and C are stronger bound together, which locks the primer in place really securely. And then extension at 72 degrees Celsius with Taq polymerase. Basically, this is Taq polymerase, a heat-tolerant enzyme from a bacterium that lives in hot springs. So it's heat-tolerant and will be able to tolerate a lot of heat. It doesn't fall apart at 96 degrees Celsius. Instead, it builds a new complementary strand starting from each primer that we had in the annealing stage. And then you repeat this cycle so many times. This is cycled over and over so many times, these 3 steps, that about 20 to 30 times, that you double the DNA so you're able to replicate it. So why does this matter? PCR is why a tiny crime scene DNA sample or a single COVID nasal swab can be amplified into enough material to actually test. So remember, there were COVID swabs that you could take. In that simple little thing that you had to do to test your nose for COVID, they can use this to be able to replicate the DNA to be able to test it. Alright, so that was a review of all the lab techniques. So now we're going to go back to the bio section, biochem. So you can also analyze proteins. The structure primarily determined by X-ray crystallography after the protein is isolated, although NMR can also be used, which is another method. And then amino acid sequence, you could use Edman degradation. For finding the concentration of protein, you can use it colorimetrically, either by UV spectroscopy or through a color change reaction. So Bradford assay, BSA assay, Lurie reagent, each test for protein and have different advantages and disadvantages, so you should study those up a little bit. Beer-Lambert law. is this equation here, where the extinction coefficient times the C, which is the concentration, times the length, will give you the absorbance. Okay, so basically the Beer-Lambert law is to find the absorption of light by solution directly proportional to concentration of the solute and the path of the length of light through the solution. And so the absorbance is how much light the sample takes in, and you can use this formula to find that, to determine the concentration of substances in the solution. So how much light that solution absorbs is directly proportional to its concentration. So absorbance is proportional to C in this formula, concentration. So if you have high absorbance, then you have a high concentration. If you have low absorption, you have a low concentration of the proteins. All right, we can move on now to carbohydrate structure and functions. Okay, so now we can go over biochem for carbohydrate structure and function. So carbohydrates can be monosaccharides, or they can be complex carbohydrates such as disaccharides and polysaccharides. So think of monosaccharides as only one carbohydrate unit, di is two, and it's due to glycosidic bonding between two monosaccharides, right? So if you have two of these guys, two monosaccharides, and you get a disaccharide. And then more than that is polysaccharides formed by repeated mono or polysaccharide glycosidic bonding. So they're all the monosaccharides come together via the glycosidic bonds. And so it would help to know organic chemistry because these diagrams will look a little bit more understandable, but you can just think of these as like molecules for now connected to each other. So yeah, in classification for carbohydrates, it would help to know organic chemistry because for the nomenclature, you have 3 carbons, which is triosis, 4 carbons is tetrosis, and etc. So you have names for them, and there's also common names, which you have to know. And you don't need to do organic chem to do that, but it will help you with understanding D and L, enantiomers, and diastereomers. So for like organic chemistry review, remember I said that things can have a handedness. So if it's L, it has a left-handedness, and then D is for right-handedness. And so if you look at the number of chiral carbons, nearly all carbohydrates in nature are in D configuration compared to amino acids, which are found in L configuration. This is confederation, that's a typo. Yeah, and so remember, carbs mostly D configuration, amino acids mostly L configuration. All right, enantiomers are stereoisomers, so versions of the carb molecules that are carb structures that are non-superimposable mirror images of the other. So D and L forms of the same sugar, so versions of the same sugar. If you don't know what enantiomers and stereoisomers are, it's good to review organic chem. And diastereomers, any stereoisomer that is not an enantiomer, so those can be epimers or anomers. And so an epimer is a subtype of diastereomers that differ at exactly one chiral carbon, a subtype of epimers that differ at the anomeric carbon. So this diagram is showing you that exactly. And then for monosaccharides, we know that you can undergo oxidation reduction, esterification, and glucoside formation. This is what a monosaccharide can undergo. Aldoses are oxidized into aldonic acids and reduced to alditols. Sugar as a reducing agent. Sugars that can be oxidized are reducing agents themselves and can be detected by reacting with Tollens or Benedict's reagents. This is important here. Tollens or Benedict's reagents. So understand this. And then deoxysugars, H replaces OH. So it's literally in the name. If you have a deoxyribose sugar, for example, instead of having OH, it gets deoxy. So minus the O will give you H. So H will replace the OH, right? Esterification, sugars react with carboxylic acids and their derivatives, forming esters. So esters, you would know what esters are from carboxylic acids and esters from your basic organic chemistry review. Phosphorylation is when a phosphate ester is formed by transferring a phosphate group from ATP onto the sugar. This reaction is similar to esterification. Glycoside formation is the basis building complex for carbohydrates and requires an anomeric carbon to link to another sugar. And so for cyclic sugar molecules, it's possible to have them look cyclic like this, like a pentagon almost, right? And so you can have them look like this, or you can have them look linear like this. And so cyclization describes the ring formation of carbohydrates from their straight chain form. So here you're looking at a straight chain form, and these ones all down here we're looking at rings, or the cyclic structure. So the anomeric carbon is a new chiral center formed in the ring closure. It was the carbon containing the carbono in the straight chain. And so alpha anomers have the OH on the anomeric carbon trans to the free CH2OH group. Beta anomers have the OH on the anomeric carbon cis to the free CH2OH group. And Hofworth projections are just able to represent the 3D structure of a monosaccharide. So something like that. Right? So this is just a way to show it. And so the mutual rotation spontaneous shift from one anomeric form to another with the straight form as the intermediate. Okay, in complex carbohydrates, they said you can have disaccharides and monosaccharides, which are basically monosaccharides times 2 plus, right? So they're bonding by glycosidic bonds. And common examples of disaccharides that you should know is sucrose, lactose, and maltose, which are all here. These are disaccharides. And then polysaccharides formed by glycosidic bonding. So these are examples of polysaccharides: cellulose and starches. Cellulose is a structural component for plant cell walls, and the main source is fiber in the human diet. And then starches are main energy source form for plants. So amylose is unbranched and amylopectin is a branched. So amylose and amylopectin are the types of starches. And then you also have to know for like the maltose here, lactose and sucrose, you also have to know like how are they bonding together and what is it. So maltose is basically glucose monomer with a glucose monomer, and they bond by an alpha glycosidic bond, whereas lactose is 2 monomers as well, because we're talking about disaccharides, so it's always 2 monomers of 2 monosaccharides. So lactose is a galactose plus glucose with a beta glycosidic bond, and then sucrose is glucose plus fructose with an alpha glycosidic bond. So there you go. And then for lipid structure and function, so now we're talking about lipids. So we went through carbohydrates, and now you're going to go over lipids. So lipids are insoluble in water and soluble in non-polar organic solvents. So think about lipids like oil or fat. Remember, you learned in basic like science before is like you can't mix oil and water because water is polar and oil is non-polar, so they don't want to deal with each other. So because oil is going to be hydrophobic, so it's insoluble in water. And then phospholipids are amphipathic and form bilayer of membranes. So if you ever heard of your lipid bilayer of the cell. I don't know if there's a picture here. Here's a phospholipid where you have a polar head. Polar head will be hydrophilic, and the nonpolar tails, which are hydrophobic. The head is attached by a phosphodiester linkage. There, that's how it's attached and determines the function of the phospholipid. So that is the phospholipid. Saturation of the fatty acid tails determines the fluidity of the membrane. So if you have a very saturated fatty acid, you'll have less fluid. And so it's more rigid. Phospholipids that contain a glycerol backbone. And then spinjolipids, this is a typo, it's not lipids, it's lipids, contain a spingelosine backbone. Many but not all spingelosines are also phospholipids with the phosphodiester bonds, and those are called spingophospholipids. Spingomyelins are a major class of them and contain a phosphadiecholine or this is a very long name, diethylamine head group, and it's part of myelin sheath. So myelin sheath relates back to biology it was about the nervous system and how neurons have myelin sheath on their axon. So if you review biology, that would be helpful. Glycosphingolipids attach to sugar moieties instead of phosphate groups. Cerebral sites have 1 sugar connected and global sites have 2 or more. So cerebral sites is important to know, and global sites. So they're types of glycosphingolipids. And then gangliosides contain oligosaccharides with at least 1 terminal N called NANA. One terminal NANA, which is N-acetylneuramic acid. Waxes contain long fatty chain acids esterified to long chain alcohols used as protection against evaporation and parasites in plants and animals. So here's an example of the cerebroside. Cerebroside was a type of glycolipid, and any lipid linked to a sugar is a glycolipid. So as you can see, this is the fatty acid unit. So lipid. And then you have the sugar unit. Sugar. What's sugar? Right? So anything with lipid plus sugar. And then here's your schematic of a phospholipid. Many use glycerol or spingosine for the backbone. So as you can see, this is the backbone using glycerol, but it can also you can also have a spingosine instead. Spingosine as the backbone. And the reason why it's backbone is carrying everything else that is here. So it has these fatty acid tails and then also this phosphate kind of head. Okay, now we can go on to signaling lipids. So terpenes are steroid precursors made from isoprene. One terpene is called monoterpene and contains 2 isoprene units. Terpenoids are derived from terpenes via oxygenation or backbone rearrangement, and they're odorous. Steroids contain 3 cyclohexane and 1 cyclopentane ring. So this is what the steroid looks like. Here's the 3 cyclohexane, cyclo for like hexane, plus cyclopentane. Here's pentane, 1, 2, 3, 4, 5. There's only 1 cyclopentane. And now steroid hormones have high affinity receptors and work at low concentrations, and they affect gene expression and metabolism. Cholesterol is important to membrane fluidity. So remember, your cells have a membrane made up of these phospholipids, or we can show the picture of this here. Right, so your cell membrane is made up of these phospholipids. And so cholesterol might be inside of a membrane, not inside of the phospholipid itself, but within the membrane to impact the membrane's fluidity and stability, and serves as a precursor to many other molecules. Prostaglandins are autocrine and paracrine signaling molecules that regulate cyclic AMP levels. So they can affect your smooth muscle contraction, body temperature, sleep wake, cycle, fever, and pain. And then you have fat-soluble vitamins, A, D, E, and K. You just have to remember that A, D, E, and K are fat-soluble, and so they like to be with lipids. They like to be with fat kind of thing. And so vitamin A function is good for is carotene, good for vision, cholecalciferol, good for bone formation, vitamin D, vitamin A, tocopherols are antioxidants, and then K, phylloquinone and metahenones forms prothrombin, which is a clotting factor, so you need vitamin K. So vitamin K includes these ones, phylloquinone and metahenones. Okay, so memorize that. Energy storage now. So you always have a way to store energy. A lot of people say fat is bad and they want to be a low body fat percentage. For example, if you're bodybuilding and you want to be super low body fat percentage, but the thing is you need energy stored in you. And so we store energy through fat too, right? So triacylglycerols are storage form of fatty acids, contain one glycerol attached to 3 fatty acid chains through ester bonds. Very hydrophobic, so do not carry additional water weight. So here's the structure of a triglycerol. So here are the 3 fatty acids, and they're connected by ester bonds. If you don't know ester bonds, again, that's from organic chemistry. Adipocytes are animal cells used specifically for storage of large triglycerol deposits. They're basically cells, fat cells. Free fatty acids, unesterified fatty acids that travel in the bloodstream, salts of free fatty acids are soaps. Saponification. So how is soap made? The ester hydrolysis of triglycerols using a strong base like sodium or KOH. So you can make salt via the process of saponification. So saponification is a chemical reaction where fats or oils, triglycerides, react with a strong base to produce glycerol and fatty acid metal salts, which is commonly known as soap. So that's how we get our soap. How is soap made, right? Micelle can dissolve a lipid-soluble, so you learn saponification with micelle because a micelle can dissolve lipid-soluble molecule in its fatty acid core and washes away with water. So this is explaining how when we do saponification, we can output soap, which is basically salt and glycerin. So remember that saponification is basically you're making soap by mixing fat with a strong base. And so micelles are a round shape that these soap molecules come in. And this shape, because of the shape, it's able to trap dirt and grease within it. And then because of the structure of the micelle, so I'll show what the micelle looks like. This is pretty good. Let me see if I can get a better picture here. So because you could see here that you have oil that's and then water on the outside. And so you can trap oils, like if you ever notice your hands greasy and then you want to put soap to remove that grease or, clean off things. This is going to be trapped inside of here. And you'll notice the micelle looks very similar to your cell membrane, which is made up of many of those phospholipids. And as I said, cholesterol could be within it, right? And cholesterol helps with membrane fluidity. And yeah, so you learn these together, saponification and micelle. Okay, now we can go into DNA and biotechnology. So we're going to learn this in a second, but I'm going to take a break myself, so see you guys in a bit. So remember we just did lipids and lipid structure and function. I actually think it's important to not follow the textbook right now and go straight to chapter 8 we were in chapter, where was lipids? Lipids, I think, was chapter 5. So I'm going straight to 8 because it has a lot more to do with lipids than the other ones, because the other 2 are about DNA and RNA, but we'll get to that in a second. So your biological membrane, fluid mosaic model. So we have a fluid mosaic model of your cell's membrane. So if you know in biology, chapter 1, we all have cells, right? All living organisms have cells, and those cells have membranes. And so, which is like the outer layer of the cell, basically. And it counts for this model. What it looks like this. And we have, as you can see, the phospholipids that make up the membrane. And it's actually a double phospholipid layer. So you'll see that you have phospholipid looking this way, in this direction, and the other layer is this way. In a micelle, for saponification is different. So the soap particles look different. So you have only one layer, and then you have the fatty layer on the inside that traps grease and dirt inside of here, inside. But this one is a lipid bilayer, so it's a bit different. As it says here, you have a phospholipid bilayer, and each phospholipid you already know that it has a hydrophilic head and a hydrophobic tail. Remember, the tail is always hydrophobic because those are your fatty acid tails. Remember that fat doesn't like water. Oil doesn't like water, for example. They're arranged so the heads are facing outward, and the tails make up the inside of the membrane. So on this inside here, you only have the tails, and that's very similar to how the micelle is structured too. And then you have lipid rafts, where lipids can move freely in the plane of the membrane and can assemble into lipid rafts. And then flipases, specific proteins that maintain the bidirectional transport of the lipids between layers of the phospholipid bilayer. And then proteins and carbohydrates may also move within the membrane, but are slowed by their relatively large size. So remember that proteins and carbs are pretty large. So that's why they move slower within the membrane. Okay, and so remember we just reviewed cholesterol. That cholesterol could be within your cellular membrane. So that's why I wanted to go over this chapter first on the biological membrane, because it is made up of lipids, really, and a lot of other proteins. That help with transportation of stuff going in and out. So the membrane components, as you know, are lipids. And we already went over what lipids are. Triacylglycerols and fatty acids, we already went over what those are. Glycerol phospholipids replace one fatty acid with a phosphate group, which is often linked to other hydrophilic groups. Cholesterol, remember we said that it contributes to the membrane's fluidity earlier in the chapter 5, I think. Was it chapter 5? Yeah, I think it was chapter 5, right, on lipids. So cholesterol, when the temperature is low, cholesterol will help increase fluidity. When the temperature is high, decreases the membrane fluidity. So helps in this balancing act for the membrane's fluidity. Waxes are present in very small amounts, and they're mostly prevalent in plants and function in waterproofing and defense. Transmembrane proteins. So there are proteins all the way up to here. These are all proteins that can be found in your membrane. So the transmembrane protein is right here. It's also known as an integral protein, type of integral protein that spans the entire membrane, and they are often glycoproteins. So that, remember, that is a conjugate protein. We reviewed that earlier. And then embedded proteins are most likely part of catalytic complexes or involved in cellular communication. So you can have any type of embedded protein within the membrane. And then you have membrane-associated proteins, which act as recognition molecules or enzymes. Now you can have also carbohydrates, which can form a protective glycoprotein code and also function in cell recognition. You can also have ligands. Extracellular ligands can bind to the membrane receptors, which function as channels or enzymes in second messenger pathways. There's also junctions. And so if you remember in the biology review, cell-to-cell communication is what we're talking about here. How can cells communicate? Actually, no. This is something else. This is related to CAM and how cells can stick together. So how are cells sticking together? So gap junctions, this is for cell-to-cell communication, where it allows rapid exchange of ions and other small molecules between adjacent cells. So that's cell-to-cell communication, but tight junctions are different. So this has to do with making sure that the cell kind of sticks together, preventing solutes from leaking into the space between the para-extracellular route, but do not provide intracellular transport. So tight junctions and gap junctions. Desmosomes and hemiadesmosomes. Desmosomes bind adjacent cells by anchoring to their cytoskeletons. So these can help with these ones can help with the structure and helping maintain the structure. And hemidesmosomes are very similar, but their main function is to attach epithelial cells to underlying structure. So these are really important for structural reasons. And also tight junctions are important for structural reasons. Gap junctions is more like for communicational reasons between cell to cell. For membrane transport, so things have to go in and out of your cell, right? So there's different ways that things can come in and out of your cell. You can have concentration gradients where things just naturally diffuse, spontaneously diffuse, in other words. And all transmembrane movement is based on these concentration gradients. And the gradients will tell us whether the process is active or passive. So if a process follows its gradient, it's passive, right? If it doesn't, then we need to use an active form of transportation. So osmotic pressure is a colligative property. So this formula is just chemistry review. Osmotic pressure is the sucking pressure in which a solution is drawing water in. And this formula is chemistry review. Passive transport does not require energy because the molecule is moving down its concentration gradient. So if you follow your concentration gradient, then it's passive. And so example of passive transport is simple diffusion. It's a form of passive transport. Small nonpolar molecules passively move from an area of high concentration to an area of low concentration until equilibrium is reached. Osmosis is basically simple diffusion, but for water, that's how I like to think of it, describes the diffusion of water across a selectively permeable membrane. Facilitated diffusion is also another form of passive transport, but you're also using these transport proteins to move impermeable salts across the membrane, so you're getting a little bit of help, which is why it's called facilitated diffusion. Okay, now we can move on to active transport, which is different. So you can draw a big red line in between passive transport, which is all of this, and then now we're into active transport, which is here. So active transport requires energy. So cells have energy in the form of ATP, adenosine triphosphate, or an existing favorable ion gradient. So you can also have diffusion, which relies on concentration gradients, or you can have ionic gradients, which is a gradient of charge. It has to do with charges. So those are two different gradients types you can have, right? Ionic or concentration, and they can happen simultaneously. So a primary active transport uses ATP or another energy molecule to directly power the transport molecules across the membrane. And remember, it's moving against the concentration gradient, if you're using active transport. And so secondary active transport, also known as coupled transport, uses energy released by one particle going down its electrochemical gradient. So when people say electrochemical, they're meaning that it's considering both the ion gradient and the concentration gradient. So that's what electrochemical is, to drive a different particle up its gradient. It's going up, or in other words, against the gradient. So if you're going against something, it's going to need energy. And so a symport is a type of secondary active transport where both particles flow in the same direction, but antiport is when the particles flow in opposite direction. So it's better to look at an image of symport. Everything's better with an image, to be honest. So following through with a structured textbook, if you're really confused, will help because they have dedicated images and explanations. So again, symport is in the same direction, but antiport is in opposite directions. Okay, and then endocytosis and exocytosis is another way, which is basically how things can move in and out of the cell. So your cell can undergo endocytosis by eating up material. So remember that this is related to the biology section on the immune system. So if you go back to biology, review the immune system, you will have a phagocytosis, which is when your body has phagocytes I don't know if you remembered that from biology, but you have phagocytes, which can do phagocytosis by eating up larger solid materials. And your body can do that when, it wants to remove, for example, something bad. So that's why it's related to your immune system. So if you go back to the biology review, it can help understand that. There's stuff on my board here. And then penocytosis is the ingestion of liquids. So it's basically drinking liquids via vesicles. Methods of engulfing material into cells or releasing. So when you're releasing, that's exocytosis, not endo. Exocytosis is whenever a cell releases material out of the cell. So I'm just going to show a picture between endocytosis versus exocytosis. So here you have exocytosis, this vesicles leaving. Endocytosis, we're taking something in. All right, and then you have specialized membranes. One second. So this is not really the best title to name this. I would only name this part specialized membranes when we talk about mitochondrial membranes. But for membranes, any membrane would have resting membrane potential. And most cells have a resting membrane potential between -40 and -80 millivolts. Normally we say -70 millivolts for an average cell or whatever. And then the membrane potential is also denoted by Vm, and it's maintained by a sodium-potassium pump as well as leak channels. So this is important. The Na plus and K plus pump and leak channels are able to make sure that your cell's membrane is at its resting potential, which is a certain number. And so the Nernst equation is related because the electrical potential created by one ion can be calculated using the Nernst equation. So this is how you would use the equation. And then the Goldman-Hodgkin-Katz voltage equation is a resting potential of a membrane at physiological temp, which can be calculated using that equation. It is derived from the Nernst equation. So you want to do practice problems to help you understand that better a little bit. So this is just about membranes. I don't know what this is here. I would name this probably, if I were writing this, I'd probably name it membrane calculations or something, calculations or equations, something like that. And then mitochondrial membranes, this I would name it as a special membrane. Because mitochondrial membranes, their outer mitochondrial membrane is highly permeable to metabolic molecules and small proteins, but inner mitochondrial membrane surrounds the mitochondrial matrix where the citric acid cycle produces electrons and are used in the electron transport chain. ETC means electron transport chain. The inner mitochondrial membrane does not contain cholesterol, so no cholesterol. So that's mitochondria. Mitochondria, remember in biology 1, the mitochondria has a inner and outer membrane. And so these are the special properties of it. All right, so we're gonna go over nucleic acid structure now, and basically what DNA is. So we'll start with what DNA is. So DNA, what is it? DNA is a long polymer made up of. So we'll start with DNA first. DNA is a long polymer. It's also called deoxynucleic acid, or deoxyribonucleic acid. Remember that deoxy meant instead of OH, instead of that OH in the structure you get H. We reviewed that earlier. And it's called a macromolecule. It's known as one of the macromolecules that stores genetic information. So DNA stores genetic information. And so they're made up of repeating units called nucleotides. Nucleotides. So this is a nucleotide. And so the nucleotide, each nucleotide has 3 parts. It has a sugar, which is called a deoxyribose. It has a phosphate group and a nitrogenous base. So I don't know if it shows it here. No, it doesn't. So I'll just show a structure of DNA. DNA molecule. Oh, this one is good. Okay, how do I bring this down? There we go. So you can see the phosphate, sugar, and the nitrogenous base. That's what one DNA looks like. Now that is a nucleotide. Now that's not just DNA, because DNA is very, very big, right? So I said, oh, that's one DNA. Sorry, I meant that was one nucleotide. And so nucleotides can be adenine, thymine, guanine, cytosine, uracil. It's basically a nucleoside with a 1 to 3 phosphate groups added. Nucleotides in DNA contain deoxyribose. In RNA, they only contain ribose. So ribose is not deoxy, right? So it has that oxygen. Okay, and so in RNA, U replaces T, so A pairs with U via 2 H bonds. So by the way, A, T, G, and U, they bond via hydrogen bonds. We reviewed that a little bit earlier. And so you can think of DNA as a string of beads, and each bead is a nucleotide, and the string itself is the backbone, and is made up of sugar phosphate links. And the decorations on each bead hanging down are the nitrogenous bases. And you have millions of these beads in a row, which make up the macromolecule of DNA. And a nucleoside is a 5-carbon sugar with a nitrogenous base, but no phosphate groups. So it doesn't have that phosphate group, as we saw in the image from before. So there are 2 types of bases, right? So you can have purines and pyrimidines, so nitrogenous bases. You can have nitrogenous bases. You can have purines or pyrimidines. Adenine or guanine are made of 2 rings, but cytosine, thymine, and uracil are made of 1 ring. So maybe I can help show a picture of nitrogenous bases to help show that ring structure. So as you can see, adenine is 2, guanine is 2, cytosine is 1, 1, and 1 for U and T, so uracil and thymine. So that was good. Now, basically, purines are like big license plates. When you have 2 rings, you have 2 sections. Pyrimidines are like small license plates. You have 1 ring, it's just 1 section. So DNA always pairs a big plate with a small license plate. So what happens is one purine will pair with one pyrimidine, and they pair together. And why does this matter? It's because this one-to-one pairing with purine and pyrimidine keeps the double DNA helix a consistent width. It's why this model, Watson-Crick model, works. Okay, so now we're going to get into the Watson-Crick model, which is, it looks like this. Watson-Crick model looks like this. It looks like a twisted ladder here. Let's just show it this way. So Wikipedia has this nice little simple image. 1 second. So this is what the Watson and Crick model looks like. And now, so the Watson and Crick model is like a twisted ladder. And so DNA isn't just a single strand of those beads. It's 2 strands wrapped around each other in a spiral way with the bases. So I should probably just keep showing the picture with the bases on the other. And this pairing follows strict rules. So A pairs with T only. So that's why you see that here. A and T only pair together, and G and C pair together. A and T pair together with 2 hydrogen bonds, and G and C pair together with 3 hydrogen bonds. And if we remember from chem bonding, general chemistry on bonding, which is like chapter 3, I think, those are pretty strong bonds. And G and C have 1 more hydrogen bond than A and T bonding together, which means they have a stronger hold to each other. So G and C is a stronger bond. Analogy is that you can think of it as a twisted ladder, and the sides of the ladder are the sugar phosphate backbones, and the rungs are the base pairs. So as you can see, these are the base pairs. And so the rungs always have the same width, purine paired with pyrimidine, as we know. So the ladder doesn't wobble or twist unevenly, and the bases are glued together via those hydrogen bonds and weak glue, so they can be pulled apart if needed, but strong enough to hold the structure together. So they're hydrogen bonds. And then this relates to Chargaff's rule, which is that in any DNA sample, the amount of adenine equals thymine, and the amount of guanine equals cysteine, which is what it says here. And so you automatically know how much T there would be, right? If you knew, for example, how much A there was, right? Because they're equal. That's just an example. And then so when you want to do DNA replication during synthesis phase, let me just go up to DNA replication. During synthesis phase, right, of the cell cycle, cells make an exact copy of their DNA. So this is related to biology when we're talking about cellular reproduction, or reproduction in that unit on reproduction, and the cell cycle. So cell cycle. So basically, during the synthesis phase part of that cell cycle, cells make an exact copy, and the double helix will then unzip, and each strand acts as a template. So new nucleotides are added to each template strand, building 2 more complementary strands. So you're building more strands. And the result is that you have 2 identical DNA molecules. So it's basically photocopying your DNA. So identical replica of DNA molecule. So that is what DNA replication is. And now for DNA repair, and also you have, what's it called? You also have DNA polymerase. Where's DNA polymerase? They didn't really talk about it here, but we might see it in the appendix later. But you have DNA polymerase, which is the enzyme that does the copying. So you should memorize this enzyme because it's important. So yeah, and this matters because this is how cells pass on genetic information to daughter cells. So it's very related to the cell cycle and mitosis/meiosis processes in that chapter. Okay, and so DNA repair is fixing mistakes. So you know, remember in the, I think, chapter 12, where you can have mutations, right? In biology, you can have mutations. And so DNA polymerase makes about 1 error per billion nucleotides. So it does make errors. And so when it's building, it needs like repair mechanisms to make sure it's correct. So these are the 4 repair mechanisms that can happen. First is proofreading. So DNA polymerase will then proofread its work. So that's why I need to talk about DNA polymerase. Well, proofread its work. And so as a DNA polymerase adds nucleotides, it checks, is this correct, basically. And then a mismatch repair over here is where you use genes to, basically, this is after you've replicated in the G2 phase of the cell cycle. And you use these genes, and basically what you're doing is that you're scanning the newly replicated DNA for mismatches. So any mismatch is just cut out and then resynthesized. So it's cutting out all the bad stuff, but that's only during the G2 phase after replication has occurred. Nucleotide excision repair is basically it's going to fix any helix lesion. So it targets damage that bends or distorts the DNA helix, like thymine dimers from UV exposure. So you know that if you don't, all the dermatologists say, if you don't wear sunscreen, oh, it's UV damage, DNA damage, whatever. That's what it's talking about. So nucleotide excision repair is able to target that damage and basically cut out that damaged section. And then DNA polymerases will fill in that gap, and it works like a cut and patch system. So that's why they said cut and patch. The excision endonuclease is the enzyme that does this. does the cutting and patching, or nucleotide excision repair, in other words. And the base excision repair is where it targets damage that doesn't obviously bend the helix, but instead what it targets is different types of lesions. So they can be called non-deforming lesions, in other words, and AP endonuclease is the enzyme that does this, and it removes the damaged base, leaving an empty spot. So it leaves an empty spot, and then the gap is filled in then correctly. So that's what endonuclease is doing there. So this is ways your DNA gets repaired. Now, moving to oncogenes, what are they? Oncogenes are like genes derived from normal growth promoting genes. So a mutation can then turn them on permanently, and that can lead to cancer. And so the reason why is because when a mutation turns them on permanently, it's like a gas pedal, and so the cell growth just doesn't stop. So you keep growing the cells, and they keep cycling, going through the cell cycle. This is why it's highly related to the biology section. Oncogenes equals stepping on the gas pedal. So you keep stepping on the gas pedal, keeps trying to replicate cells. And so that leads, and that's a very bad thing. And tumor suppressor genes are code for proteins that slow down or stop cell cycling. So it's the opposite, reduce. And so when these are mutated, it's like cutting out the brake so that the cell can't stop growing. So it just can't stop anymore. So an analogy is that a normal cell is like a car with a gas pedal and brakes, right? But proto-oncogenes are like the gas that grows, and then the tumor suppressors are like the brakes that stop cell growth. If the gas gets stuck on an oncogene mutation or the brakes fail with tumor suppressor mutation, then the car crashes. Cancer usually requires both of these to happen, so both of this and this can lead to cancer. So the cell keeps growing forever, and why it matters is because it explains why cancer requires multiple mutations and why some people inherit a higher cancer risk because they're born with one of these systems already weakened. So that is the part on DNA repair. And now we're going on to chromosome organization. So what is it? In eukaryotes, DNA is wrapped around histone proteins. So DNA doesn't just travel by itself. It travels being wrapped around these histone proteins. Let's see, histone proteins to form nucleosomes. So these nucleosomes are further coiled and looped to form chromatin. They form chromatin. And the chromatin is then organized into chromosomes. So chromatin is organized into chromosomes. I don't know, they didn't put it here. But you know that chromosome is like this guy. And so now we can understand that there are two types of chromatin, that there is hetero and euchromatin. And hetero is, as it says, dark, dense, silent, and euchromatin is light, uncondensed, and expressed. So what does it mean to be silent versus expressed? So silent means that the genes are turned off, but expressed means that the genes are turned on. Some special features that you should know about chromosomes is that they have telomeres and centromeres. So telomeres are at the end of your chromosomes, and centromeres are just in the middle. So I'll show a picture of chromosome. Here we go. This is a good picture. So telomeres at the end and the centromeres in the middle, as you can see. And the agrocentric chromosome is basically what it says here. When the centromere is located near one end of the chromosome and not in the middle. All right, so that was eukaryotic chromosome organization. It's just explaining what the chromosome structure is. And also you have 46 chromosomes in human cells in total. What else do we need to go over now? The other ones. So we will do that. Let's go over biotechnology and tools and how DNA can be manipulated. So scientists use three main tools to work with DNA. So they can have recombinant DNA and hybridization, polymerase PCR, so polymerase chain reaction, or electrophoresis. So we already went over electrophoresis, I believe. So you can separate DNA by size. You can also amplify a specific DNA sequence. So we already went over that. These are both in the lab techniques that we just went over. So check mark for both of those. For recombinant DNA, you can take DNA from two different sources. And then what you could do is hybridization. So it would be the joining together of the complementary base pair sequences. So complementary DNA strands are mixed, heat denatured, and allowed to reanneal and come back together. And if they're complementary, they will stick together. And this is how you detect certain DNA sequences. All right. And then we can go into nucleic acid structure. Nucleic acid structure are nucleotides. So we already went over nucleotides, but this is just a specific structure about them. So you can have, as well, primary, secondary, tertiary, and quaternary. And so nucleic acids are polymers of nucleotides, and these include DNA and RNA. So your primary structure is just a linear sequence. Secondary structure, so these are proteins. That's why it follows the same thing as like looking at protein structures. Interactions between bases with the same molecule. The bases are held together by hydrogen bonds. Secondary structure is responsible for the shape of nucleic acid. And these are the four basic elements. They have loops, helices, bulges, and junctions. Loops include stem loops, also known as hairpin loops, tetraloops, and pseudoknots. In the tertiary structure, the location of atoms are in 3D space now, very similar to the way protein is in primary, secondary, tertiary, and quaternary. interactions of nucleic acids with other molecules, such as chromatin interacting with histones, is when you get quaternary. So as you can see, DNA here, it has histone protein. So that would be this chapter on DNA. Okay, so now we can go over, we went over DNA, and now we're going to go over RNA, but more than that, the bigger picture of all of this RNA and genetic code is that DNA to RNA can make protein. And we'll see how the cell does that in a second, and we call this the central dogma. Central dogma. And that's what it says here. Central dogma states that DNA is transcribed to RNA, which can be transcribed to protein. In other words, they can answer the question, how do our cells make protein? Yeah, protein. So an analogy is to think of it like a cookbook in a library. The cookbook is your DNA. It tells you and it gives your cell kind of like instructions. And when you need to cook something, you then photocopy that specific recipe and you can make it into, let's say, RNA. And you take that photocopy to your kitchen and you hand it to your chef, who's your ribosome. Ribosomes are organelles. If you don't remember from biology chapter 1 on cell review, is an organelle that will make protein. And that's the chef. And the chef reads it and makes that dish. So it's reading the DNA instruction to make then protein. Well, the instruction that originally came from DNA but became transcribed into RNA, and then it's reading RNA to then make protein. So that is the central dogma, and that is the main thing about the whole chapter. That's why it goes over translation, transcription. So the genetic code is basically a dictionary that translates the 4-letter alphabet, the DNA bases A, T, G, and C, into a 20-letter alphabet, which are the amino acids. Remember, you have 20 amino acids, right? So the code reads in triplets called codons. So let's say you have A, T, C, G repeating in whatever way that, for example, I'm just writing random stuff. And basically, how do you read this, right? How do you read that? Well, you read it every 3 bases. So 3 of these will code for 1 amino acid. And we remember amino acids are making up proteins, right? 3 will read into another amino acid, specific type. So that's how it works there. So you have a degenerate code. So the G-generic code is that most amino acids are coded by more than one codon. For example, UCU and UCC code for serine. So it doesn't just have one codon. So one of these is a codon, right? Codon for an amino acid. But amino acids usually just don't have one. And then start and stop codons, they're basically AUG. A start codon is always going to be AUG. So AUG is always a start codon. And that is in the name. It always starts the reading frame, and it basically signals, hey, we're starting to read from this point. We're starting to read the instructions at this point. But it also codes for methionine, which again is amino acid. And then stop codons are UAA, UAG, and UGA. People like to memorize the stop codons, these ones, by you are annoying, you go away, you are gone. That's like a mnemonic to help someone remember. And then point mutations are different damage, different consequences. So every point mutation has different types of damage: silent, nonsense, and missense. It's another biology review from chapter 12. Silent mutations have no effect on protein synthesis, usually found on the 3rd base of the codon. Nonsense mutations that produce a premature stop codon, so any one of these. And then missense are mutations that produce a codon that codes for different amino acids, like entirely different amino acid gets coded. I should zoom in so it's easier to read this. And then frame shift mutations is when the entire frame is literally shifted, as it says. So frame shift happens when the nucleotides are added or deleted, but there's no swapping, which shifts the entire frame. And the ribosome reads codons in groups of 3. So if you delete one base, now it's reading the wrong 3 triplets the whole time. So where was my fake sequence? Here's my fake sequence, right? So if I deleted A, then all of a sudden it shifts the entire frame, and now I have to read it like this, you know? So that's what happens in a frame shift. And then RNA, for RNA, it says it's similar to DNA except ribose sugar instead of D-ribose, uracil instead of thymine, single-stranded. This is review. And then 3 types of RNA include mRNA, tRNA, and and rRNA. So basically what happens is that for messenger RNA, it's transcribed from DNA in the nucleus, and it travels to the cytoplasm for translation, which is a process explained here. And so tRNA is able to bring amino acids and recognizes the codon on the mRNA using its anticodon. rRNA makes up the ribosome and is enzymatically active. So this is the 3 types of RNA, they become more clear once you start learning about translation. But it might still be confusing to you if you don't know about translation. And then another thing to note is, wobble. So the 3rd base in the codon allows for mutations to occur without effects in the protein. And that can lead to that is the reason why silent mutation can occur, because the 3rd base could be whatever the heck it wants to be without changing the actual protein. And wobble base pairings are less stable. So where was my thing on that? I had a little note for it that I wanted to add. I guess it's a little bit gone now, but now we can go into translation and how it fits with what we were just talking about. So what is translation? So translation is like translating your DNA to RNA. right? And so when you translate, this is where the mRNA code gets read and turned in to protein. So the ribosome is the shelf, and it reads the mRNA codons and adds, I actually think, one second. So let's go back to the central dogma, because I think it'd be too confusing to go right into translation, because we should learn transcription before translation. So this is the overall process. We're using DNA to turn into RNA, and that RNA will be used to turn into proteins. So, sorry. So what is transcription? We'll start there. So transcription is the process where the cell will copy a section of DNA into RNA, and the DNA stays safe in the nucleus, and the RNA is just temporary, and it's a portable copy of that DNA that gets sent out. So remember in biology 1, you have a nucleus, and that holds your DNA, right? But the RNA is like the portable version of your DNA. And so it sends instructions out to the factory floor where the proteins can get made. So in transcription, you're unzipping the DNA. So remember we had a double helix DNA structure, and basically this exposes the template strand that could be copied. And so the RNA polymerase, RNA polymerase is important. RNA polymerase 2 finds this promoter region. It's called a promoter region, which is the start here signal. So let's see if here, DNA polymerase 2 finds a promoter region, right? And what it's going to do is it's going to start there and signal on the DNA. In eukaryotes, it looks for the TATA box. TATA box. So if you go up here, in eukaryotes, it looks for this TATA box within that region. And so a specific region of 25 base pairs of where transcription should start. So this region is about 25 base pairs. Okay? And it says, let's start copying from this point on. Okay. And now what's going to happen is you're going to have the building of the RNA strand from DNA. So this enzyme will now start building RNA. So it reads the template strand, remember, from unzipping the DNA, and builds a complement strand. So you remember that DNA got unzipped. Now it looks all parallel like this. And now it's going to use one of these sides, template, to build a complement, to build a complementary RNA strand. Complementary. What does that mean? Well, let's say DNA, you have A, T, G, and C. A will pair with RNA U, that's complementary, U. This one will pair with A, G will pair with C, and C will pair with G. So let's do a better picture of transcription. I saw a good one by, like, Khan Academy had a good one too. Let's see. This one's okay, I guess. Here's a picture. So you can see here that you have that RNA polymerase, DNA that encodes. Okay, well, I don't know why we're talking about rho factor right now. I just want a simple diagram here. So RNA polymerase, and then it's making mRNA from that template strand here. So it's using one of the sides. And I unwinded this DNA. As you can see, it's not winded anymore on this part right here. And these are 25 base pairs. Remember that, right? And so the raw copy, the HNRNA, is the RNA that comes directly off of this. So what that picture was showing is building it, right? And that would be called your the raw copy would be called the hRNA. hRNA, right? So it still has junk in it. It's not yet. It's still a messy copy. That's why that picture is a little bit misleading. I know it says mRNA for this one, but really what it made is hRNA. And that is before any edits have happened. Because again, you know, your polymerases can make mistakes while they're building stuff, right? So it can make mistakes, and the raw output we get is hRNA. But when we get an edit, it edits this. So it edits it. We call it post-transcriptional modification. Post-transcriptional modification. And this is the editing process, and it's only done by eukaryotes only. So that's when you get exons and introns. So we get exons and introns and post-transcriptional modification. So remember, we have hRNA, the collective term for unprocessed mRNA that happens. So yes, it is, hRNA is technically mRNA, but it's mRNA that hasn't been yet edited. So yes, we have hRNA that's going to go through post-transcriptional modifications. And those include, actually we should go over exons and introns. So before we can use hRNA, it needs to be edited. And so exons are express sequences, and these code for amino acids. They exit the nucleus and become part of the mRNA. They will form part of the mRNA. Whereas introns are going to be spliced out. These are things in, for example, you're an editor for a movie scene. You want to split out a certain scene that wasn't entertaining or good. Yeah, your introns are going to be like that. The parts that need to be spliced out so they stay in the nucleus of the cell. And so introns also enable alternative splicing. What's alternative splicing? Alternative splicing is like the director's cut. So here's where it gets really smart, because sometimes some of the raw footage can be edited in different ways. So maybe scene 3 is kept in one version but cut from another. So we have different versions of scene 3. Or scene 7 is inserted in one edit but not another. So the same gene or raw footage can be produced in multiple different mRNAs. So we can end up with multiple different mRNA versions. That's why it's called alternative splicing. So a certain exon may be cut out or an intron may stay. This allows for RNA segment to code for more than one gene. So this is why one gene can code for multiple proteins. Okay, so the modifications that can, what are the exact modifications that can be made? We know what, can be staying, which is exons, and what can be removed, which is introns. We know about alternative splicing, which is different ways we can cut the things. But now we need to know what would the post edits be, other post edits be. And in that case, we can have a 5' cap, or you can have a poly A tail. I think they just wrote it here. 5' cap and poly A tail are added to mRNA. The cap tail stabilize for translation. So we're trying to prepare this for translation that will happen later on, where we turn RNA to protein. Now, prokaryotic cells can increase variability of gene products from one transcript for this polycistronic genes, and these are multiple translation sites within the gene which lead to different gene products. So basically, in eukaryotes, we have this modification of the 5' cap and the poly A tail. What is the 5' cap? It's a modified guanosine nucleotide added to the beginning. It's literally like a bottle cap, right? Like a cap. And it protects the mRNA from being degraded and helps the ribosome recognize its real mRNA. So it's going to help later on for translation. And then poly A tail is a long string of adenine nucleotides, such as A, right? Adenine added together at the end. And this is like a cap before the end. This is like a cap. 5' cap is like the cap before the start. So poly A tail is exactly the name. It's an A tail. That's at the end. It's a tail added to the end, and this protects the mRNA and helps it stick longer. So just imagine the mRNA or the hRNA has then it's hRNA, but then if I add the post-transcriptional modifications, so I'm going to add the cap, and then I'm going to add the AA tail, and that one is right here. And that's after I have these post-transcriptional modifications done. And now that I have hRNA turned into mRNA, I can then send that out for a process of transcription to then make proteins. And who's making the proteins again? The ribosomes. Yeah. Okay, so to explain the prokaryotic thing, prokaryotes have no nucleus and no introns, so their transcription and translation happens at the same time. That's basically what happens. So they have these two processes happen at once, and so they can make something called polycistronic mRNA, and one mRNA codes for multiple proteins. So one of these guys can already code for multiple proteins, and they do these two steps together for prokaryotes, so they're much more simpler. But we have another step here to go over, which is translation. So translation is basically where the mRNA gets read and converted into protein, and a ribosome reads those codons. So remember, we made mRNA with the reading strand and codons and stuff. Codons 3, 6, 9, etc. So they have 3 base codes. In other words, 3 base codes. And for each codon, a tRNA will deliver a matching amino acid. So tRNA is important now. tRNA translates the codon into the correct amino acid. And then now we have ribosomes. So tRNA will bring in an amino acid based on that 3 base code. And so, or that instruction, right? And the amino acids link together in a protein chain. So this will link, this one will be linked, this one will be linked. And once you get enough amino acids, you're making a protein, right? Etc. So then you have the ribosome, which is the factory where translation or protein synthesis occurs. This is where it makes the proteins. And inside the ribosome, you have different sites. You have the P site, you have an A site, and you have an E site. And each site does a different job. So in your P site, this is where the tRNA hangs out with the growing peptide chain. That's growing peptide chain. That's what we just saw earlier. And the A site is where the arriving tRNA, when new amino acids will dock. So new incoming amino acids go here. It's a very simplified version of review, by the way. And the E site is where any empty tRNAs will end up leaving. tRNAs leave. So in eukaryotes, we have bigger ribosomes. We have 80S ribosomes, meaning 80, I think, subunits, right? 70S, these are smaller. So what happens is you can think of tRNA as delivery trucks, and tRNA is a molecule shaped like a clover, and one end has an anticodon which matches the base with an mRNA, and the other end carries an amino acid. So it has 2 places. It has a place, kind of how the structure of tRNA works is it has a structure that could read this codon, this 3-letter code, and then it also has a structure that can carry an amino acid with it to bring over. And where is it bringing over? It's bringing over to the ribosome. So yes, your mRNA is like your instruction tape. And so the 3 stages of translation follow this. So in prokaryotes, it's gonna be hard without a picture. I think I'm gonna show a picture as well. Or no, we'll just follow this. So the first stage is initiation. Initiation is basically where you're starting the machine. So in prokaryotes, the small ribosomal unit, which is 30S here, attaches and scans for the mRNA looking for the shine delorno sequence, the specific sequence, and scans for the start codon. And then it lays down this N-formal methionine, whatever. So what it needs to find is the start codon. So remember, the start codon over here is AUG. So it's looking for that code. And it attaches, and special tRNA carrying this, tRNA would carry this in the P site. And then it would dock in the P site. And then the large subunit, 50S. So remember, prokaryotes total have 70S, so they have a 30S subunit here, 30S, and then 50S subunit attaches, completing the ribosome. So that's how it works. The large subunit then attaches completing the ribosome. And then for eukaryotes, you have a small subunit, 40S, which attaches to the 5' cap. Remember, we just learned about that, 40S, and scans for the start codon. Remember, AUG. It lays down methionine, that amino acid, in the P site of the ribosome. So methionine docks in the P site, and the large subunit attaches completing the ribosome. In stage 2, you have elongation. So elongation is where you're adding amino acids one by one. That's what it means by elongation in this one. So it's a repetitive cycle, and it happens over and over until the protein is built. So codon recognition is what happens first, and the ribosome exposes the codon in the A site. So remember, the A site was over here. The ribosome exposes the codon in the A site, and a tRNA with a matching anticodon docks in the A site. So the tRNA will dock here with the amino acid that it's going to bring in. So then it's going to dock there, and it ends up like doing peptide bond formation. So the amino acid in the P site, part of the growing protein chain, bonds to that A site. So the amino acid that is in the P site, so remember, this is the growing strand, so this last one here, for instance, is going to then bond to the incoming amino acid that was brought by tRNA in the A site. So A, you can imagine A for arrival kind of thing. And then you have translocation, which is where the ribosome shifts one codon forward. So what happens is that the empty tRNA in the P site moves now to the E site. So there's an empty tRNA that just gave away its amino acid, right? So then it's going to move to the exit site. And then in the exit site, it's going to leave, literally. The tRNA with the growing chain now in the A site moves to the P site. So it's just a big shifting. And now the A site is empty and ready for the next tRNA. And tRNA keeps coming in with its amino acid until this protein is finally built. And it repeats the steps, peptide bond formation, translocation, over and over again. And then termination is when we finally stop. We finally stop the process. So you need to reach a stop codon, right? A stopping code. So remember the mnemonic, you are annoying, you go away, you are gone, right? So that can help you remember them. And so when you hit the stop, when the ribosome hits the stop codon, and it's like, oh, we finally reached, on this code here, you are annoying. We reached finally the stop codon. Okay, so it knows that now. And what it's going to do is no tRNA has an anticodon for stop codon. So what happens is that a release factor is a certain protein that recognizes the stop codon and docks in the A site. So it's not the tRNA that finds this out and can read this stop codons. It's the release factor. So that's why we have down here release factor. So now that the release factor has read it, it catalyzes the addition of a water molecule to the end of the protein chain, and this breaks the bond holding the protein to tRNA. So water is able to break the bond between tRNA and the protein. And now the protein can be released. It's done. And then you finally just get your protein, releases the protein. And then let's just say that the whole process of translation doesn't happen perfectly either. You're going to have, so in transcription we had after that post-transcriptional modifications. Now in translation, you're also going to have what we call post-translational modifications. And the reason why is because, you know, our cells are not perfect. They're like workers. They make mistakes. So what are the finishing touches? Well, there can be folding that happens by chaperones here, and they help the protein fold into its 3D shape, formation of quaternary structure, 3D shape, quaternary structure. And then cleavage is some proteins have signals, sequences that get cut off. Cleavage. Sometimes things get cut out, you know. And then covalent addition or chemical addition is when the protein might get phosphorylated. You can go through carboxylation, glycosylation, or prenylation. So phosphorylated means you get phosphate group added to it. Glycosylated means you get a sugar added to it. And prenylate is a lipid added to it. So prelinated here, it's a lipid. Sugar added. This is carbohydrate added, I think. And this one is phosphate group added. Yeah. DNA ligase is basically... it's exactly what, fuse the DNA strands together to create one strand. So it's able to fuse a lot of DNA together by forming phosphodiester bonds between sugar phosphate backbones of DNA fragments. And so you can think of DNA ligase as like a stapler for DNA, right? So that's DNA ligase. All right. So now we can go over control of gene expression for both prokaryotes, so prokaryotes over here, and then eukaryotes. So now we're going to go over gene expression. So why does control matter? So a cell has a lot of genes, like 20,000 plus or whatever, 30,000, whatever, it has a lot. But it doesn't need to use all of them all at the same time. So a liver cell doesn't need eye color genes all the time, right? So all our cells, the important key concept is all our cells technically have the same genes, same DNA, but we've selected for certain ones that are on and certain ones that are off. And this is what gene expression control is. So the question it answers is, how does the cell decide which genes it wants to use? How does a muscle cell know that it wants to only use specific genes to be a muscle cell, for instance? So in prokaryotes, it follows this model called Jacob-Monod model. Let's do a highlighter. Jacob-Monod model, and explains how operons work. So an operon is a cluster of genes that work together to perform one job. They are clusters of genes that are trying to perform one job. They're all transcribed together as one mRNA, and they're controlled by a single off and on switch. In inducible systems, the lac operon is an example of it. Inducible systems, the default state is just off. So they're bonded to a repressor, and the default state is off. So what happens is that when it's turned on, an inducer, the substrate, the genes will then process. Okay, and then we have repressible systems, which is basically where the default state is actually on. So the default state would be on, example, TRP operon, and then it's turned off by a co-repressor, the end product that's usually very abundant, so co-repressor. All right, and now we can go onto how do eukaryotes have gene expression. So we have transcription factors. So what are transcription factors? Actually, let me explain why eukaryotes have a little bit more complex control than prokaryotes. So eukaryotes are much more complex because we have a nucleus where we have also a separation between transcription and translation, as I said before. But we also have chromatin, which is where DNA is wrapped around histones, and we have multiple layers of control that happen. So the first layer we can think of is transcription factors. So proteins that bind to specific DNA sequences can either turn the genes on or off. So they bind to the DNA and they can turn things on or off. Promoters. So where do those proteins bind? Promoters are within 25 base pairs of the transcription start site, as it says here, and enhancers are more than 25 away from the transcription start site. So you can think of promoters as the ignition, and that's where the RNA polymerase initially binds. Enhancers, they boost transcription from a distance, and they can allow thousands of base pairs away to do this, but they still affect the gene through DNA looping. So another thing to highlight is chromatin and chromatin structure. So remember that earlier, DNA is wrapped around histone proteins. So I'll try to show DNA and histone. I'll just try to show a picture of that so it's more clear. I don't really like these pictures that I'm seeing right now. Here, we can use this one. So DNA is wrapped around a histone, or you can look at this one. There's the histone, and the DNA is just wrapped around it like a rope kind of thing. And so basically, this packaging has a very it has a purpose. So what one of its purposes is to control access to genes. So if the DNA is tightly wrapped or condensed, or it has transcription factors and RNA polymerase, right? So if it has these stuff, and this is tightly wrapped around the histone, the genes are not accessible. So decreases accessibility, or you can have increases of accessibility if the DNA is loosely wrapped around the histone. So transcription factors can now access the gene, and the gene is now on. So this affects basically accessibility. So a histone acetylation, this is important, is a process where acetyl groups are added to histones, and the histones then loosen their grip on DNA, and the DNA then becomes more accessible. For DNA methylation, this process is important to review. It's the exact opposite. So methyl groups are added instead directly to DNA bases, especially to cytosine, and they act like a do not disturb sign. So transcription factors can't bind anymore. So they're saying, no, don't come in, no. So it closes off access. And this is called heterochromatin. Heterochromatin. I didn't write it here, but heterochromatin. And so the complete picture is that for eukaryotic gene to be expressed, there are three things that need to happen. Three things to express gene in the eukaryote. So the first thing is that the transcription factors must be present. They have to be there. You need transcription factors. The second thing is that these factors need to bind to promoters and enhancers / enhancers. And the third thing is that chromatin must be open or accessible. So you need histone acetylation to help you with that, to loosen it, not methylation. That process will tighten it and reduce accessibility. Remember that? So if any of these fail, right? If any of these are failing, then you won't be able to turn on the gene. So then it wouldn't be expressed. All right, so we are now on chapter 9, 10, 11, and 12. And so for chapter 9 and 10, they kind of go together, and they're under the branch of learning about carbohydrate metabolism, which is very long because there's many different processes that can be involved in carbohydrate metabolism. And then also there is then chapter 11 with lipid and amino acid metabolism, which is a way that our bodies can turn lipids and amino acids into energy. For the carbohydrate metabolism, it's very long because there's so many different things and processes involved in turning our carbohydrates into energy, and there's different pathways in which we can do that too, so that's why it's pretty long. And then for chapter 11, just to give an overview, it's about lipid. Okay, we already went over that. And then chapter 12 is about bioenergetics and regulation of the metabolism in general, so making sure everything is balanced. So they all go together, these last 4 chapters. So the first thing I want to go over is that our body is like an economy in a sense, and so instead of teaching 9 different separate pathways, I wanted to do like a basic overview in terms of learning it like an economy or like a city. So our body is kind of like the city, where we have one economy, and every term, I'm just going to lower the size, every term in all 4 of these handouts, or the 4 chapters that I showed earlier, have a role in the single story. And if students hold on to this map that is shown below, we can start to understand more. So I'll show you the map in a second. So the running analogy is this. It says to write on the board and leave it up. I'm not writing it. I literally copy and pasted it, and it's a good analogy made by the artificial intelligence. So your body runs a city's energies economy, and food is like the income. You need food. Your body, our bodies need food going in. Food goes in. Now, what comes with our body's processes is it's able to then use the food income. Food is our income, but you can't pay anything anymore directly. What your body needs to do is to turn that food into energy, cellular energy known as ATP. ATP is that energy that your cells need. It's a form of energy that your cells need. ATP is very important. And everything else in metabolism is either earning it, storing it, or spending it. So ATP. So that's our body, a cell's energy, right? Main form of energy. And so cash is like glucose, or glucose is like cash. Basically, it's spendable by anyone, but it's very bulky to store glucose in your body. But it's good. You can spend it as a currency. ATP is a mid-level energy made and spent constantly, and it's never hoarded. So your body's always using this ATP, and it's the only accepted currency in the cell. And now we have NADH and FADH and NADPH. And these are like trucks or transportation for high energy electrons to cash in and get, in the end, ATP. Eventually, our goal in the end eventually is to get enough ATP. And a power plant, it's known as the power plant, or the powerhouse of the cell. It's also known as the powerhouse of the cell, is the mitochondria. And so that's where trucks unload, and most of the ATP is actually made. It says minted, but it's made. So your ATP is made there. And so in the checking account, right, your checking account is your savings in real life. And in your body also has savings in terms of energy. We can save our energy with glycogen. Fast access, small capacity, and lasts about a day. We can also save energy by triacylglycerol in adipose, those fat cells. So our fat cells contain a lot of energy for us long term. It has enormous capacity, and it's slow to access. Our body also has body protein. You can only burn it when you're desperate and under starvation. You also have your liver. Your liver is the only organ that can put glucose back into circulation, and it's the only organ that can do that. Insulin is when you have a fed state, anabolism, and you start to store everything. And then when you have glucagon, epinephrine, and cortisol, you have a fasting, stressful state, catabolism. So you are basically, catabolism is destroying, and then anabolism is building. So you're storing stuff, building stuff, and then this is releasing everything energy-wise. Think of all of this as energy-wise. Metabolism has to do with energy. Energy, that's the main part. And now ketone bodies is what the liver uses when the cash runs out. So when you have no more energy, no energy, well, what is, your brain needs some energy somewhere, or else you'll go brain dead and die. So what the liver is going to do is going to get ketone bodies. So that's your last resort. So the way I would actually teach this is to look at not firstly the glucose transport or any of the processes here. I would look at a bigger overview first, which is our biogenetics and regulation of our metabolism, which is actually in the last chapter. So if you look at this, we have ways to help our body know like spending and spending energy, how it's going to convert energy, and like how it's going to have it all in equilibrium. So again, our bodies have these metabolic states. So we just talked about the economy. Our body's like an economy that wants to have a certain equilibrium, is an undesirable state for most biochemical reactions, because organisms need to harness free energy to survive. So you don't actually want to really be in equilibrium, but the thing is, you're always going to be in not equilibrium for biochemical reactions. Not EQ for biochemical reactions. So we usually think of equilibrium as we want to be in it, but you notice that when you're you have different states in your body. So you have postprandial state. So I'll teach you off of this, where you're well fed and your insulin is going up. So anabolism happens, right? And then post-absorbent state is when you're fasting. And so well fed, by the way, is like you just ate a meal. And then post-absorbent state is you're fasting. And so your insulin will lower. These are hormones, and your glucagon will go up, and catecholamine as well. And so it transitions to catabolism, where we're breaking things down. For example, breaking down a molecule of food that will be able to then give us energy, or for example, breaking down fat to be able to give us energy. So the main thing is that we're breaking things down to give us then energy somehow, right? And so prolonged fasting is when you are basically starving, and that increases glucagon and catecholamine. And then most tissues rely on fatty acids. So 2/3 of your brain can be derived from ketone bodies. All right, so what's important to remember is that carbs, fats, and proteins all will lead down to one road. So we know we can get all these macromolecules from our food, right? And so they all lead down to one road, and that is acetyl-CoA. acetyl-CoA. It all leads down to acetyl-CoA. So if we go back to our handout, because I still want to follow the handout so it's easy to follow along, acetyl-CoA is the first thing that's talked about in chapter 10, and acetyl-CoA contains high energy bond that can be used to drive other reactions when hydrolysis occurs. That's the basic definition. Now we're going to go into bioenergetics, which is in chapter 12, so that you can better understand more about the body and how it works. Okay, so before you ever learn about how energy is moving at all, like all these processes and all the other chapters, like I said, we need to go over like how does this energy move at all? Why is energy moving in our body at all? And that's bioenergetics. So in an open system, we have matter and energy, which can be exchanged with the environment. So you eating and breathing. You eating and breathing. That's matter and energy exchanged. You know, like I'm eating, so I'm putting matter in my mouth, right? Food is matter, and I'm putting it in my mouth. A closed system is only energy can be exchanged, so no work is performed because pressure and volume stay constant. So this equation applies here. Delta enthalpy is equal to delta internal energy and heat exchange. If these terms, by the way, are very confusing, then you need to review general chemistry on systems and processes. Okay, so the next thing, so general chemistry review would help a lot for thermodynamics, which goes over systems and processes. And then the next thing you need to know is that in entropy, it's just a measure of how dispersed energy is in your system. So a lot of dispersion means high entropy. Not a lot of dispersion, low entropy. You can think of it as like diffusion of energy. It's very diffused. That's how I think of it personally, and it works for me, but dispersed works as well. Free energy can come in two versions, and so we have change in free energy. So we can have standard free energy, which is your delta Gibbs free energy that you learn in general chemistry, and the energy change that occurs at 1 mole concentration, 1 atm, 25 degrees Celsius. So you notice in general chemistry, that would be the, I'm pretty sure, STP conditions. So, actually, sorry, these are standard conditions. Standard conditions, my bad, because it's in standard state. So this is standard conditions. Standard conditions are thermodynamic standard state here is when your temperature is at 25 degrees Celsius, 1 molar for all solutions, and it says 1 bar here, but we are going with 1 atm. Okay, so just know that these are basically standard. So that's why anything delta G, anything with a circle there means standard for that variable. That's why it has that little degree symbol there for delta G. And so modified standard state is another one, and you see that it has this little prime. That means modified. See that little quotation mark, the single quotation mark? And what it does is it's the same thing as this, but it's corrected to your physiological conditions. So same thing as standard free energy, but the difference is that it's corrected to your conditions, which is a pH of 7. So the body's pH of 7. H plus concentration is equal to 10 to negative 7 molar. All right, and then we have ATP. Right, so we talk about ATP a lot. It's a mid-level energy molecule, and this is what ATP is your energy, your cell's energy. And it's not necessarily the most energetic molecule in the cell. It's deliberately mid-level, and it's high enough to drive most reactions, but low enough that plenty of molecules can recharge or use it. So ATP hydrolysis is around this level. So it's not the most highest source of energy, but it's in this medium range, mid-level. And so it does have these high energy phosphate bonds. So there's phosphate in it. I believe it's right on there. You can't really see it that well. There's an ATP structure there, diagram there. So what is that bond high energy? What is that? Because the products are stabilized after hydrolysis by 3 things. So the reason why the bond energy is high is because we get resonance, ionization, and the loss of charge repulsion. So resonance is the released phosphate spreads its charge over more atoms. Ionization is the products ionize favorably at cellular pH, and loss of charge repulsion is that the stacked negative phosphates were pushed. We're pushing each other apart, splitting them. And so, like ATP does this job in 2 ways. So there's hydrolysis and coupling. And so you would get hydrolysis coupled essentially to an energetically unfavorable reaction. And then you can also have phosphoryl group transfer, where you donate a phosphate to another group. So it's like transferring the phosphate to another group. And you'll see the second one immediately in glycolysis, where ATP can hand a phosphate to glucose to make glucose 6-phosphate. So there is such thing as phosphoryl group transfers, but we'll see that later in action. So again, ATP is like your $20 bill for the cell, or it's like the cell's energy currency. And so it's not a gold bar where it has like a huge amount of energy within it, or it doesn't have huge delta G free energy. Instead, it's about mid-range, right? And it's high enough to buy almost anything that the cell needs, but low enough that dozens of processes can do something. And so the cell chose a currency enough, that's just enough. And so that is ATP. We have a currency of ATP, which is your cell's energy. So now, for biological oxidation and reduction, in this one, we see that redox reactions can be broken into half reactions. So this is important to know, because what you realize is that in general chemistry, if you have a redox reaction, so you can essentially split this reaction into an oxidation reaction, so iron oxidation, and then this other reaction is the reduction of it. So what's iron doing? They're writing it out over here, and what's chromium, I think this is chromium, doing over here. And so that's how in chemistry, I'm not doing a review in chemistry right now, but redox reactions can be broken down into what I just showed you, half reactions. So half reactions means one half is going to be the oxidation half, and the other half is going to show the reduction reaction. And basically, by the way, a redox happens, the reason why we even say redox instead of just oxidation and then reduction is because they basically happen at the same time. Alright, so we know that, and that can give you information about more about stoichiometry and thermodynamics and the molecules around the high energy electron carriers. So remember, we introduced the high electron carriers, NADP, FADH, ubiquinone, cytochromes, and glutathione, and NADH here. So all of these are able to carry high energy electron. That's why we call them high energy electron carriers. And flavoproteins are a subclass of riboflavin vitamin B12. And in other words, you can name them as FAD or FMN. And you'll meet FMN again later on. So all of these are playing roles in your biological redox reactions. If you don't know redox reactions, then you're going to need to go back and review Gen Chem. So a lot of this is general chemistry, right? So that's how general chemistry relates to your body biology, and that's why I guess it's called biochemistry. And we already went over the metabolic states, so that's great. Put a check mark on that. And so if a question ever gives you a delta G prime, where was it? Over here. Instead of this one, in standard condition, it's telling you what's happening in physiological conditions, in other words, conditions in your body. And if it asks you why oxygen is the terminal electron acceptor, it's because it has the highest reduction potential. That's how it relates to this redox over here. If something has a high reduction potential, if something has a high reduction potential, it means a chemical species has a strong positive tendency to gain electrons and undergo reduction. These substances act as strong oxidizing agents because they pull electrons away from other molecules. So they have electron gain. A high positive voltage means the reaction favors taking electrons. So essentially, if you have a high reduction potential, that's why oxygen can be a terminal electron acceptor, because it wants to accept that electron. Oxygen really wants to accept that electron because it has the highest reduction potential. All right. So we went over this. Now we can go over glucose transport. So all this is good for now. I think we can move on to glucose. So we're gonna, where's the stop button? Glucose in our terms can be basically like sugars. And we eat sugar a lot in our diet, right? So how does glucose transport within your body? Well, we have something called GLUT2 and GLUT4. So GLUT2 and GLUT4. GLUT2 is found in your liver for glucose storage, and pancreatic beta islet. So if you don't know what that is, you need to review biology a little bit. Cells as part of the glucose sensor and has a high Km. So GLUT2, where is it found, right? It's found in your liver and also the beta cells in the pancreas. It's high Km, meaning it has a low affinity, right? What it means is that it only takes up glucose when blood glucose is actually high. So it acts as a sensor, as part of a glucose sensor. GLUT4, where is it found? Adipose tissue and muscle, okay? And it will have a low affinity. So if it has a low affinity, what will happen? It will grab glucose even at low concentrations. So it's not stimulated. GLUT2 is not stimulated by insulin, but GLUT4 is. Insulin. It will respond to insulin. So you can think of GLUT2 as a flood gauge on the riverbank. It sits high up, so it only gets wet when the river is genuinely high. So genuinely high meaning you're high in blood glucose. You have a lot of glucose in your system, like sugar, for example, right? Which is exactly what a sensor should do. That's why pancreas uses it, GLUT2. So the beta cell needs to know, is our blood sugar actually high before releasing insulin? And so GLUT4 is like a bucket brigade that only forms when the foreman blows the whistle. In other words, GLUT4 are the buckets are great at scooping stuff, and they stay in the shed until insulin calls them out to the membrane. So they want to be called out by insulin. And that's the muscle and fat. They don't take glucose on their own initiative. They take it when they're told by insulin. That's why they have to be stimulated by insulin. And so a high Km would mean it has low affinity, so it only works when the substrate is abundant. And so a sensor only works when the substrate is abundant, which is, glucose. So these are basically our glucose transport system here. So I wanted to show a picture of GLUT2 as a channel. Oh, whoops. Oh, gosh. I don't know where it went now. Here's GLUT2. Where's GLUT4 now? I need to find GLUT4. Okay, Okay, but anyways, GLU4 looks similar, so just know that. So we know glucose could be processed, right? But what about other monosaccharides like galactose or fructose, right? So galactose comes from lactose in the milk, and it's trapped by galactose kinase. And then it's converted to galactose 1-phosphate via galactose 1-phosphate orotransferase. So this is just a big name for an enzyme that's able to convert 1-phosphate, convert this 1-phosphate. And also epimerimerase helps out too. So these two are working together, these two enzymes, there's two of them, are able to convert it to galactose 1-phosphate via those two. So this galactose can be able to convert to 1-phosphate, right? And then for fructose, like something where it comes from, like honey, fruit, and milk, you're eating that type of stuff, or even processed food, sometimes I see it like has fructose in it, it's trapped in the cell by fructokinase. So fructokinase traps that. And then it's cleaved or cut, or cropped, in other words, by aldose B to form glycerolide and DHAP. So an analogy is that phosphorylating a sugar is like stamping the bill. So once the bill is stamped, it can't leave the building. The membrane transporters won't recognize it anymore. So every sugar that enters the cell gets stamped for exactly this reason, trapping. Glucose gets stamped by hexokinase. So remember, glucose gets stamped by hexokinase. And then galactose gets stamped, as we saw, by galactose kinase, right? And then fructose for fructose kinase. And so they need that same move for all the three sugars. It's just there. So your body is just trying to keep sugar within the cell, basically, okay? And it maintains that inward flow of nutrients. So now we want to break this income flow of energy, of glucose, and we want to break it down. And so what does that breakdown look like? So glucose plus 2 NAD plus 2 ADP plus 2 phosphate leads to 2 pyruvate plus 2 ATP plus 2 NADH and 2 hydrogen ions. So glycolysis happens, again, in the cytoplasm. So it needs no oxygen. No oxygen is needed, okay? And that's a modest of 2 ATP. So as you can see, there's 2 ATP that's being formed here. 2 ATP. And the small number is the point. Glycolysis isn't where we get all the ATP. It's a small amount. It's not that much, unfortunately. So the first thing that happens is we need to look at a diagram of how glycolysis works. So here's a diagram of glycolysis. So you have 3 irreversible enzymes, and this is 6 of the 10 glycolysis steps are like freely reversible. 6 out of 10 of them, and 3 of them are not. And those 3 are where a lot of the regulation happens. But there's a lot happening in glycolysis. I think we can go to the meltdown sheet actually, so that people could follow along better. Glycolysis. So you got to remember basically all of the steps. So in the first step, you get glucose that turns into glucose 6-phosphate. With hexokinase being the helper there, and that's what it's doing. And then glucose 6-phosphate turns into fructose 6-phosphate with the help of this enzyme, phosphoglucose isomerase. And then with the help of and then from fructose 6-phosphate turns into fructose 1,6-biphosphate. And this can actually turn into dihydroxyacetone acetone phosphate to glyceraldehyde 3-phosphate. And we have these enzymes here again to help out. And this enzyme does for this part, this conversion here. This enzyme for converting this, so 1,3-biphosphoglycerate, and then this enzyme, this kinase, helps with converting to this. This enzyme helps for converting to this, 2-phosphoglycerate, and then from there we get PAP, and this enzyme finally helps with that, turning into pyruvate. And so the fate of pyruvate is this. Where can we go with pyruvate? So we've done all the steps of glycolysis. Now what happens? All these 10 steps are important to remember. 2, 3, 4, 5, 6, 7, 8, 9, 10. The fate of pyruvate can go into the Cori cycle, where it's turned into lactate, and that will end up making lactic acid, which is not something you really want, really. And then citric acid cycle, acetyl-CoA, it can make acetyl-CoA, which can be led to the citric acid cycle, which is what you want generally for this pathway is cellular respiration. And now acetaldehyde, pyruvate can also become acetaldehyde and become ethanol. All right. So then this is why we have this diagram for the fate of pyruvate. So PAP is high energy because the phospho group traps PAP in its enol form. So enol is a part of organic chemistry review. If you don't know what enol is, that's okay for now. Just know it's enol form when the phosphor group is donated to ATP, making ATP, so the enol converts to a more stable ketone pyruvate, which releases a lot of energy. So this is PAP here. And so your final process of glycolysis will give you that equation we were looking at earlier. So let's first highlight, the 3, so there are 3 irreversible enzymes that are highlighted here. Hexokinase, PFK, and then you have pyruvate kinase. So basically, for hexokinase, it's trapping the sugar, So it wants to trap the sugar in the cell because we don't want the sugar going anywhere else, so we're going to use it. And then PFK is when there is no point of return. That's why it says committed step. No point of return. And so, yeah, we're moving forward from there. And then pyruvate kinase is where substrate level phosphorylation can happen. Substrate level phosphorylation can happen. Okay, so that's good. So in the first half, what's happening from, let's do a different color then. From glucokinase and hexokinase, they're both irreversible. So hexokinase and glucokinase are irreversible. So what I wanted to show from that is that they're both able to convert glucose to glucose 6-phosphate. And the difference is the location and personality. So glucokinase, remember, was in the liver and pancreatic beta cells as part of the glucose sensor, remember GLUT2. And then hexokinase is part of peripheral tissues. So it's like a worker who stops when their inbox is full. So it's inhibited by its own product, which is GCP, which is over GCP. Give me G6P, right here. So it's like a classic negative feedback. That's why it says here regulated by G6P feedback. So then we also have PFK1. PFK1 is right here, which will turn the phosphofructokinase 1 phosphorylates into fructose 6-phosphate, and from fructose 6-phosphate turns into fructobiphosphate here. And so this is the rate-limiting step of glycolysis. So if you don't understand the rate-limiting step, then you need to go back to review chemistry, general chemistry, on rate-limiting steps. But this means it's the slowest, basically. So it's activated by AMP. That's why it says regulated by ATP-AMP ratio. So it's activated by AMP and fructose 2,6-biphosphate, which is here. And then it's inhibited by, so it'll stop when there's ATP, a lot of ATP. So it's like, oh, we're rich, so we need to stop. So yeah, that's ATP is regulating it. And also citrate. Yeah. And then for PFK2, the enzyme's only job is to talk to PFK1. Another enzyme I want to mention is glyceraldehyde 3-phosphate dehydrogenase. It's able to produce NADH, as you see here. And so this can feed later on into the electron transport chain, where obviously you're transporting electrons over, and NADH, as we saw before, was one of the possible electron transport carriers. So this is what the enzymes were listed out as here, and they listed out even their function, which we were going over earlier. So again, glucose kinase was irreversible, hexokinase irreversible. All of these irreversible ones are important to know. And then it says that glucokinase converts glucose to glucose 6-phosphate. In the pancreatic, beta is itself as part of glucose sensor. Hexokinase converts glucose to glucose 6 in the peripheral tissues, inhibited by G6P. We went over that. PFK is also known as phosphofructokinase 1, and it phosphorylates fructose 6-phosphate to fructose 1,6-biphosphate as a rate-limiting step. So it's the slowest step, and it's activated by AMP and fructose 2,6, but it's inhibited by ATP and citrate. So it's all here. And then phosphofructokinase 2, we said, produces 2,6-biphosphate that activates PFK1. So this activates that. This one activates that one. And so it is activated by, this one is activated by insulin and inhibited by glucagon. So these are hormones, right? And glyceraldehyde 3-phosphate dehydrogenase produces NADH, which can feed into the electron transport chain. Pyruvate kinase can perform substrate level phosphorylation. We went over that. The NADH producing glycolysis is oxidized by the mitochondrial electron transfer chain. So remember ETC? I said the NADH would then go to the ETC. But if O2 or mitochondria are absent, the NADH producing glycolysis is oxidized by the cytoplasmic lactate dehydrogenase. So it might actually be going lactate. It might be oxidized into lactate dehydrogenase. It might be oxidized by lactate dehydrogenase. Okay, so now we can go over your checking account, your body's checking account. Glycogen. Where is glycogen? Glycogen. And also now we can go into gluconeogenesis, actually, which is the opposite of what's happening. So it occurs in your cytoplasm and mitochondria, predominantly in the liver, with small contribution from the kidneys. Most glyconeogenesis is simply the reverse of glycolysis. So again, it's the reverse of what we just learned in glycolysis, and it uses the same enzyme, The 3 irreversible steps of glycolysis must be bypassed by different enzymes. So remember, these were irreversible. So it's like, how can you do glycolysis backwards if we have enzymes that are irreversible and stuff like that, or reactions that are irreversible? So how are we going to do a reverse reaction such as gluconeogenesis? Well, it's because they're bypassed by different enzymes. So different enzymes are going to help out with that. Pyruvate carboxylase converts pyruvate to oxaloacetate, which is converted to PEP by PepCK. Together, these 2 enzymes bypass pyruvate kinase. So this is our bypass. This is helping us bypass. Pyruvate carboxylase is activated by acetyl-CoA. PepCK is activated by glucagon and cortisol. Fructose 1,6-biphosphate converts fructose 1,6-biphosphate to fructose 6-phosphate, bypassing fructose. So this is another enzyme to bypass, and this is the rate-limiting step of gluconeogenesis. This is important. It is activated by ATP and glucagon, inhibited by AMP and insulin. So if we go to gluconeogenesis here, this is the opposite of this, glycolysis. Glycolysis occurs in your cytoplasm. They also give you a nice review of glucose having 6 carbons and pyruvate having 3, and what pyruvate looks like over here. And then we went over all of that. Gluconeogenesis takes place in the liver and to a lesser extent in the kidneys, but mostly in the liver. And so this is taking glucose and going to pyruvate. That's what it looks like from here. But if you learned it, we're actually going the opposite direction from pyruvate this way. That's what it really is. Gluconeogenesis is the opposite pathway, going the opposite way. This one's going down, straight down, down to pyruvate. So pyruvate in gluconeogenesis is going up this way. So basically, we go from pyruvate. We have these pyruvate carboxylase. We said PepCK was there as well. We needed that one. We needed this one, fructose 1,6 biphosphatase, glucose 6 phosphatase. So all of these ones that are highlighted in blue are all very important ones to memorize here. And same with this diagram here. And then most of the other stuff is just memorizing that pathway. Alright, so now glycogenesis and glycogenolysis. So glycogenesis is the production of glycogen, so your energy stores or your fat stores. And they use two main enzymes, glycosynthase and branching enzyme. And it occurs only in your liver and muscle cells. So glycogen is stored in your liver. And so for glycogen synthase, it creates an alpha-1,4 glycosidic bond between glucose. Remember earlier that we had a carbohydrates review, and so this is what it is. And then you have a branching enzyme, which creates branches with alpha-1,6 glycosidic bonds. So glycogen is a very big molecule thing, big thing, right? Glycogenolysis is the breakdown of glycogen, so it's the opposite. Instead of production, it's breaking down. So these are opposing each other. Using two main enzymes, glycogen phosphorylase and debranching enzyme. And you can imagine what debranching enzyme does because we already know what branching enzyme does. So debranching enzyme moves a block of glucose from a branch connected to a chain using alpha-1,4 glycosidic bond, and it removes the branch point, which is connected via an alpha-1,6 glycosidic bond, releasing the free glucose molecule. Now the glycogen phosphorylase is different. It removes the glucose 1 phosphate molecules by breaking alpha-1,4 glycosidic bonds. So this one is breaking the one that was synthesized here, and debranching is breaking the bonds that were synthesized here. So they're opposing each other. They're different. Right? So if I were to draw a line between the two, this is the line. Whoops, that's not a very nice line. But glycogenesis involves these two, and glycogenolysis involves these two enzymes. 1 and 2. Okay, so basically, glycogen is like your energy stores, or your energy bank account, or your energy account. I don't know. It's like your bank of energy. And sometimes you want to make glycogen stores like fat to store, right? And then like you have this energy stores, but sometimes you want to take that out because you need that stored energy for energy. You need to do stuff before you get too tired. So then what you're going to do is your body's going to break it down using glycogenolysis. So you need both of these processes. And then we can go over glyconeogenesis. No, we already went over that one. We're now going over pyruvate dehydrogenase here, and then we're going to go over the pentose phosphate pathway. Sorry. So in pyruvate dehydrogenase, it's where enzymes convert pyruvate to acetyl-CoA. So remember, if we go back to glycolysis, our final product is pyruvate, and we're going to go, pyruvate has different fates, but one of the main pathways is that after pyruvate is made, it turns into acetyl-CoA, which enters the citric acid cycle, also known as tricarboxylic acid cycle, also known as your Krebs cycle. So there's a lot of names for it for some reason, I don't know why, but yeah. And then, so then if we go back to metabolism 1, right, so it's able to convert pyruvate to acetyl-CoA right before the citric acid cycle. It is stimulated by insulin and inhibited by acetyl-CoA. So this is the cycle that has to be understood and memorized. So then the pentose phosphate pathway, you can think of it as, sorry, one second. You can think of it as, it's also called a hexose HMP shunt, and it occurs in the cytoplasm of most cells. And it takes almost no ATP, and it's not trying to. It doesn't want to take up energy, or take from the cell's energy. So when glucose 6-phosphate enters the pathway, and the products are NADPH sugars for biosynthesis and glycolysis intermediates. So glycolysis 6-phosphate enters the pathways, and those are the products that you get: ADPH sugars for biosynthesis and glycolysis intermediates. And the rate-limiting enzyme is glucose 6-phosphate dehydrogenase, right here, which is activated by NAD plus insulin and inhibited by NADPH. So an analogy is that you can think of glycolysis as the highway to the power plant. So glycolysis is the main way we can get glucose into your mitochondria fast. So your glucose can make pyruvate, and then you can get pyruvate to turn into acetyl-CoA, and then acetyl-CoA can enter the citric acid cycle, and then that can go to your ETC, which is now finally in your mitochondria. And as you know, the mitochondria is where all your ATP energy is. A lot of it is going to be made. So so you can think of glycolysis as that, and basically this pentose pathway, you can think of it as a side hustle almost. You don't really take it to make more ATP. You take it because you need it to build the materials and stuff like materials like ribose for DNA and RNA, or NADPH which the cell uses to neutralize like oxidative damage and build fatty acid cholesterol. So a cell that only ran on glycolysis would be rich but unable to build anything, right? So it's something that runs in parallel, right? Parallel, oh, where is it? That runs in parallel to glycolysis, right? And as I said, it matters because it's able to fight cellular stress and also support growth. And these are the two main phases that were broken down here, so you can understand more about the pentose phosphate pathway. So now we're getting into the second part of carbohydrate metabolism. So if we went through the pathway, we probably of glycolysis, then we've already made, and pyruvate dehydrogenase has done this for us too, is we've been able to make acetyl-CoA, right here, acetyl-CoA. And now that we have acetyl-CoA from these processes described, we get to here, acetyl-CoA, perfect. So it's high energy bond that used to drive other reactions when hydrolysis occurs. You can form it from fatty acids, which enter mitochondria. Fatty acid couples with CoA in the cytosol to form fatty acyl-CoA, which moves in intermembrane space. The acyl fatty acid is transferred to keratin, acyl-keratin, which cross blah blah blah. The acyl group is transferred into a mitochondrial CoA to reform fatty acyl-CoA, which undergoes beta oxidation to acetyl-CoA. So the beta oxidation is let me just show beta oxidation of fatty acid is described in this image here, as you can see. So you gotta start memorizing a lot of pathways, right? So pyruvate dehydrogenase is able to oxidate, so we were just talking about it up here, is able to oxidize pyruvate, creating CO2, and it creates thiamine, vitamin B1, and and Mg magnesium 2+. And then dihydropolyl transacrylase oxidizes the remaining 2 carbon molecules using lipoic acid and transfers resulting acetyl group to CoA, forming acetyl-CoA. Dihydropolyl dehydrogenase uses FAD. So these are different ways that they're making the so uses FAD to reoxidize lipoic acid, forming FADH2. This FADH2 can later transfer electrons to NAD+, forming NADH. Now it's an electron carrier. That's an electron carrier that can feed into the ETC. ETC is the electron transport chain here. And so pyruvate dehydrogenase kinase phosphorylates PDH when ATP or acetyl-CoA levels are high, starts turning off. So pyruvate dehydrogenase phosphatase dephosphorylates PDH when ADP levels are high, turning it on. So these are opposite. Now, basically, acetyl-CoA will then enter the citric acid cycle, which is over here. The citric acid cycle takes place in the mitochondria, basically, and its main purpose is to oxidize carbons and intermediates to CO2 and generate high-energy electron carriers. These are the ones, and GTP. So citrate synthase, conase, isocitrate dehydrogenase, all of these enzymes here, you need to understand how they're being made. This is how succinyl-CoA is made. It hydrolyzes the diacetyl bond in succinyl-CoA to form succinyl-CoSH, and this enzyme generates the one GTP generated in the cycle. So that's how GTP was generated from over here. Succinate dehydrogenase oxidizes succinate in the form of fumarate. This flavoprotein is anchored to inner mitochondrial membrane because it requires FAD, which is reduced to the form of one FAD generated in the cycle. And fumarase and malate dehydrogenase. So all of these are going to be the enzymes in the citric acid cycle, as we see soon. Go to the citric acid cycle. So the first thing is that in eukaryotes, the citric acid cycle, CAC is the abbreviation in this handout, in the mitochondria, and the prokaryotes happens in the cytoplasm. So remember that we had pyruvate dehydrogenase, which is able to turn pyruvate into acetyl-CoA, and from acetyl-CoA, you can bring that to be processed in the citric acid cycle. And glucose turning into pyruvate is done via glycolysis, which was up here, glycolysis. And so in the citric acid cycle, step 1 is you start with acetyl-CoA, and that's step 1. And then that turns into citrate, and then that turns into isocitrate, which turns into alpha-ketoglutarate, which turns into, where's that, succinyl-CoA. We have succinate, we have fumarate, malate, oxalatate, and then it goes back. So remember that it's not just taking the same thing and going back over, it's taking new incoming acetyl-CoA and then processing this way. So things to note here. Step 3 is your rate limiting step, so it's going to be the slowest step, and then these are all of the worthy enzymes to memorize and all their functions as it laid out on the review sheet. So citrate synthase, aconitase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, succinyl-CoA synthase, succinate dehydrogenase, fumarase, malate dehydrogenase. And then the most important part is understanding okay, so we had this many reactants, what products did I get from that process? And so we got one ATP, which is important, but more important than that, we had electron carriers that were produced. And these electron carriers carry high energy electrons, which can be sent to the ETC, which is the electron transport chain. And then these are the important enzymes that they highlighted in blue, or no, they didn't highlight them in blue actually, but they highlighted them by writing the step number because they were important. Step 1, right there. So let me just erase these so it's easier to see then. The important enzymes. Citrate synthase, so citrate synthase, isodehydrogenase, and then alpha ketoglutarate dehydrogenase complex over here. So step 1, 3, and 4 are important, and then it's also important to know their inhibitors and activators. What inhibits the enzymes and what activates them. Remember, they're enzymes, so they can have inhibitors, and we already talked about inhibition beforehand. Okay, so now we can go back to that same sheet. Where is it? Biochem, Biochem Metabolism 2. And so in complex, so you have your electron transport chain, which is able to take the NADH that donates electrons to the chain. So NADH will donate electrons, which are passed from one complex to the next. So complex are like compartments on the ETC, and as ETC progresses, reduction potentials increase until oxygen. Remember, we talked about oxygen. It has the highest reduction potential, so it wants to receive all those high energy electrons at the end of the chain. It's like they're playing hot potato, and oxygen gets the potato basically. And sometimes, since NADH cannot cross the inner mitochondrial membrane, you get these other two shuttle mechanisms to transfer electrons in the mitochondrial matrix. So we'll see that later as well. So now remember that the important part from the citric acid cycle was these electron carriers, NADH, FADH. So you can see now that they're going through NADH and FADH are going through this electron transport chain, and eventually what's going to happen is the cell is going to have ATP synthase, which spins, and it goes through osmosis to make a lot of ATP. And so a lot of ATP is generated via this process, and from only one glucose, you can get a total of 32 ATP from going through all the processes, not just citric acid cycle, but for glycolysis, you have to go through glycolysis, then pyruvate dehydrogenase, or the oxidation rate, and then citric acid cycle, and then you will get this total. So each NADH can make 2.5 ATP. Each FADH can make about 1.5 ATP. And all of these are also important to remember, like what processes is the glycolysis and what it makes. What does the dehydrogenase complex make? What does the citric acid cycle end up making? So one acetyl-CoA will lead to 3 NADH, 1 FADH, and 1 GTP. That's from the citric acid cycle, CTA. It writes it right there. Or sorry, CAC. My bad, CAC. And glycolysis forms 2 pyruvate. So 2 acetyl-CoA molecules will exit the PDH complex. So PDH makes 2 acetyl-CoA molecules descend to the citric acid cycle. And so a total of 6 NADH, 2 FADH2, and 2 GTP molecules happen per glucose molecule. So what you want to understand is that, I have 1 glucose, right? Then what is that 1 glucose going to make if I put it through glycolysis? It's going to turn into 2 NADH and 2 ATP, but that's not the product it actually makes, right? But what it makes after glycolysis is pyruvate, and then pyruvate can go through this complex and turn into the 2 acetyl-CoA, and then 2 acetyl-CoA can make this. When 2 cycles of the Krebs cycle will give you this result. So that was a lot of writing. I'll just delete that now. So as you can see, the electron carriers from food. So NADH is now going through complex 1, FADH is going through complex 2, and both of them are sending to Q electrons. These electrons are moving electrons through the electron transport chain. That's why it's called the electron transport chain. Complex 1 is known as this name, complex 2 has this name, complex 3 has this name, complex 4 has this name. This is complex 1, 2 is down here, 3 is here, and 4 is over here. And so now we can go back to understanding that the protein complex of electron carriers are these ones, right? So now we can successfully go back. And by the way, what happens is that these complexes pump out H+, right? And so there's so much H+ in this area that this H+ gets diffused it gets pushed back into this area. And it gets pushed back into that area. So then what happens is that's able to generate ATP via chemiosmosis. So all of this over here is considered oxidative phosphorylation. This small part is considered the electron transfer chain, and this part over here is going to be chemiosmosis. So if we go back to Metabolism 2, you have complex 1, which uses iron sulfur to transfer electrons from NADH to FMN, then to CoQ, forming CoQ-H2. So there's some other parts I didn't really show, but CoQ is there. 4 H+ ions are translocated by complex 1. Then you have complex 2. This is the name of it, and it uses iron sulfur cluster to transfer electrons. So you're still transferring electrons, but there's no pumping at complex 2. You're going to note that. There's no pumping at complex 2. And then complex 3, this is the name, remember that, we saw that earlier. So it's able to form cytochrome C as part of the Q cycle, and H+ ions are translocated by the complex 3 as well. There's H2+ ions being translocated by complex 4. So basically, to be translocated by a complex describes the physical movement of the molecules across the membrane. As you saw in the picture, you had the complex, and then you had H+ being pumped out of it, and that's translocation. So NADH cannot cross the inner mitochondrial membrane, so therefore we have shuttle mechanisms to help it cross. So you're wondering, oh, how did it cross the mitochondrial membrane? Well, we should have learned this first to understand that. We have a glycerol 3-phosphate shuttle, and we also have a malate aspartate. So electrons are transported from NADH to DAP, forming glycerol 3-phosphate. These electrons can then be transferred to mitochondrial FAT, forming FADH2. Electrons are transferred from NADH to oxaloacetate, forming malate. Malate can then cross the inner mitochondrial membrane and transfer electron to mitochondrial NADH, plus forming NADH. Alright, so that was a lot, wasn't it? This is a mechanism for acetyl-CoA to enter the mitochondrial matrix. The mitochondrial matrix is where acetyl-CoA can undergo beta oxidation to form acetyl-CoA. So this is how acetyl-CoA is formed, right? Oxidative phosphorylation. So we went over oxidative phosphorylation in the diagram image, but basically all of oxidative phosphorylation includes the proton motive force and ATP synthase. So the proton motive force, remember there was a lot of H plus being pumped out, so the complex. So when this one, the complex, it's getting a lot of this H plus translocated out of the membrane. So it creates an electrochemical gradient, as you can imagine, generated by the electron transport chain across the inner mitochondrial membrane. The intermembrane space has higher concentration of protons than the matrix, so this gradient stores energy, and this can be used to form ATP via chemiosmotic coupling. So this, using the protons that were pumped out and using that electron gradient that's formed from all that pumping, we can then make ATP basically. So the ATP synthase that you saw, the last big protein looking thing, right, on the membrane, it was like that, is responsible for generating ATP from ADP, right? And so the F0 portion has an ion channel allowing H plus to flow down the gradient. So remember that big red arrow that pointed downward, and then it was like generating ATP, right? So allows the H plus, so the H plus is what's going down the gradient from the intermembrane space to the matrix, and the F1 portion uses the energy released by the gradient to phosphorylate ADP, so it's making a lot of ADP into ATP. So that's what ATP synthase is doing. It's like in the name, it's synthesizing ATP. Alright, so we're finally entering the less heavy stuff. Now we're into lipid and amino acid metabolism. So how can we use sometimes fat, kind of, and protein as our energy sources? So usually we don't actually like to use protein as an energy source, like ketone bodies via ketogenesis, but we can. And we could also use fatty acids with fatty acid oxidation happening. So let's go through it first. So lipid digestion and absorption. So we need to eat lipids. So mechanical digestion of lipids occurs primarily in your mouth and stomach, so you're just eating them. The chemical digestion is when digestion of lipids occurs in your small intestine and is facilitated by bile, pancreatic lipids, and cholipase, and the cholesterol esterase. So emulsification is upon entry to the duodenum. So if you didn't do biology, you should go back to the digestive system chapter on literally the digestive system, because the duodenum is part of your intestine. So it's important to remember that. Duodenum. Emulsification occurs, which is the mixing of two normally immiscible liquids, in this case fat and water. So fat and water usually don't mix together, right? But this increases the surface area of the lipid, which permits the greater enzymatic interaction and processing. So the duodenum is able to do that. Emulsification is aided by bile salts. Micelles. Micelles are water-soluble spheres with a lipid-soluble interior. Remember we talked about saponification in micelles. This is a micelle. And water-soluble solid spheres, right? And their digested lipids may form micelles. When you digest lipids, they can form micelles to be carried to the intestinal epithelium, where they're absorbed across the plasma membrane. So micelles are important not just for soap, but biologically we use them, these structures. Short versus long chain fatty acids. Short chain fatty acids are absorbed across the intestine into your blood, but long chain fatty acids are absorbed as micelles and assembled into chylomicrons for the release into the lymphatic system, into the lymph system. Here's the image for it. Lipid mobilization and transport. So how do we get these lipids moving around? So lipid mobilization. Lipids are mobilized from adipocytes by hormone-sensitive lipase. Lipids are mobilized from lipoproteins by lipoprotein lipase. So I feel like it said the same thing almost twice. Chylomicrons transport dietary triacylglycerol cholesterol, cholesterol esters from intestines to tissue, and it uses the lymphatic system. Lipoproteins, the transport mechanism for lipids, very low density, delivered to tissues. Intermediate density will have transition particle VLDL to IDL to VLDL, but low density is bad, so it moves cholesterol into your tissues. High density is good, it moves your cholesterol into your liver and will exit the body. So basically you can think of lipoproteins as transport mechanism for lipids. And so lipids are mobilized from adipocytes, and lipids are also mobilized from lipoproteins, right? So yeah. And then apolipoproteins form the protein component of lipoproteins, and they are receptor molecules that control interactions between lipoproteins and the tissues that they're docked with. They didn't write anything here, but that's basically what they are. So a good analogy to think about is the lipoproteins are like delivery fleet, and apolipoproteins are the address labels on the side of the truck. And what tells a tissue this truck is for you, open the dock. So that's what the apolipoproteins are. They're like labels. And so now the direction, LDL, is the delivery truck driving cholesterol out to the neighborhoods. The neighborhoods would be your tissues, right? So that's what LDL is doing. And now, and dropping it off. And if you have too many of those, cholesterol piles up on the streets into your arteries, which is bad. Not good. HDL is a garbage truck driving the other way, collecting cholesterol from the neighborhoods and hauling it back to the liver where it could be dumped out, and that's a good thing. Good. Okay, so now we can go into fatty acids, because those are lipids. So again, you can have fatty acids that are carboxylic acids with long chain that are saturated or unsaturated. Saturated means you have no double bonds. Saturated means you have one or more double bonds. Fatty acid synthesis is synthesized in your cytoplasm from acetyl-CoA transported out of the mitochondria, and they involve 5 steps: activation, bond formation, reduction, dehydration, and a second reduction. Arachidonate is a precursor to eicosanoid signaling molecules, prostaglandins. So there's a lot of signaling molecules here, also precursor to leukotrienes. So fatty acid oxidation occurs in the mitochondria following the transport by the keratin shuttle we saw beforehand. And then we have beta oxidation, uses cycles of oxidation, hydration, oxidation, deolysis, cleavage. The fatty acid chain is shortened by 2 carbon atoms. FADH, NADH, and acetyl-CoA are then generated. So carboxylic acid alpha N versus this carbon counting starts at the other end. Yeah. And then we can go into ketone bodies, protein catabolism, and cholesterol metabolism. So we were talking about cholesterol just before, so we could do that again now. So in cholesterol metabolism, cholesterol can be obtained through dietary sources, or through de novo from your liver. Could do that. So this reductase synthesizes this mevalonate. This is a rate-limiting step of cholesterol synthesis. LCAT is catalyzing the formation of esters for transport with HDL, and CEP catalyzes the transition IDL to LDL by transferring cholesterol esters from HDL. So that is cholesterol metabolism, and that's related to this over here, so you can study them together. And then for protein catabolism is about protein digestion. So we're going to break down protein catabolism, right? So to break down protein digestion occurs in your small intestine. Catabolism of cellular protein only under conditions of starvation. So big protein we're talking about happens whenever you're eating protein, right? But the small cellular protein, we only break down that when we're starving. So remember we talked about the starving state? Yeah. And this is ketolysis. No, give me a second. Amino acids released from proteins usually lose their amino group for deamination. The resulting carbon skeleton can be used for energy. So glucogenic amino acids can be converted into glucose through gluconeogenesis, all but leucine and lysine. So remember gluconeogenesis. Ketogenic amino acids can be converted into acetyl-CoA and ketone bodies. Leucine and lysine are the only amino acids that are solely ketogenic. So these are the fates of the amino acid carbon skeleton following the protein catabolism. So you have to memorize which ones. All right, and then for ketone bodies, basically for ketone bodies, you have a traveler check. Okay, what the heck is a traveler check? So let's say that, just give me a second, I'll pull it up. So let's say that there's a problem with your body and it's in starving. That's why I related it up here. Starving. So the volt, or your fat, is full, but the brain can't spend fat. Your brain needs some other way to get energy, not fat. So what is it gonna do? And remember, they can't the blood-brain barrier, fatty acids can't cross it, and remember that they can't be turned into glucose either. So the liver does something interesting. It takes the fat it's burning and converts the excess acetyl-CoA into ketone bodies. Ketone bodies, you can think of them as traveler's checks. So they are water soluble, so they can travel in the blood like normal. And critically, the brain will then accept them. So this is basically to feed your brain, during starvation state. And so you're feeding your brain, and ketogenesis is the liver that's able to issue all of these ketone bodies formed by ketogenesis due to acetyl-CoA from the liver during a prolonged starvation state. And so for ketolysis to occur, you have to regenerate acetyl-CoA for use as energy source in peripheral tissues. In an energy source, the brain can derive 2/3 of its energy from ketone bodies during that prolonged starvation. So during prolonged starvation and everything is out, all you have left is, your brain needs energy. This is the system it's going to use, ketogenesis. All right, so I hope that's good. So remember that ketone bodies are the body's answer to a specific constraint. The brain runs on glucose and can't run on fat. So without ketones, prolonged fasting would require breaking down muscle protein for gluconeogenesis at a very deadly rate. So ketones exist to spare protein, so you're not just using up all your muscle mass. And that's why it's what makes two-thirds of its energy from ketone bodies during prolonged starvation. All right. So now we have our last but not least, we're going over the hormones that are helping regulate our metabolism. So we heard insulin a lot, glucagon, glucocorticoids, catecholamines, and thyroid hormones, and they're all working together. And then we have tissue-specific metabolism as well, and then we have integrative metabolism. So we're gonna go over that right now. So you can have insulin, which is secreted by beta cells in the pancreas, from the pancreas, regulated by glucose, low glucose by increasing cellular uptake, increased rate of anabolic metabolism. Secreted by alpha cells in the pancreas is glucagon, stimulated by low glucose and high amino acid levels. Increases blood glucose by promoting gluconeogenesis and glycogenolysis, the processes we just went over. Glucocorticoids increase blood glucose in response to stress by mobilizing fat stores and inhibiting glucose uptake. They increase the impact of glucagon and catecholamines. An example is cortisol. Catecholamines promote glycogenolysis and increase basal metabolic rate through sympathetic nervous system activity. It's like your adrenaline rush. So an example was epinephrine and norepinephrine. Your thyroid hormones increase your basal metabolic rate, evidenced by O2 consumption and heat production when it's secreted. T3 and T4. Out of both of them, T3 is more potent, but it has a shorter half-life and is available in lower concentrations in the blood. T4 is converted to T3 at the tissues, and thyroid hormones are tyrosine-based. So this is a good picture of what it's trying to summarize here. Then we have the liver, which is the most metabolically diverse tissue. So this is the liver, right? And so hepatocytes are responsible for the maintenance of blood glucose levels by glycogenolysis and gluconeogenesis in response to pancreatic hormone stimulation. The liver also processes lipids, cholesterol, bile, urea, and toxins. Adipose tissue is stored lipids, like you can think of that as fat, under the influence of insulin and releases them under the influence of epinephrine. So these are how the hormones are coming into play as well. Skeletal muscle metabolism differs depending on the activity level and the fiber type. So if you're resting, you conserve carbohydrates in glycogen stores, like fat, and use free fatty acids from your bloodstream. If you have active muscle, like if you're doing activity of some kind, you may use anaerobic metabolism or oxidative phosphorylation of glucose or direct form of phosphorylation from creatine phosphate, that's another pathway, or fatty acid oxidation depending on the fiber type and exercise duration. So think about what type of exercise a person is doing, and think about which pathway would they most likely be using. Cardiac muscle uses fatty acid oxidation in both well-fed and fasting states. Brain and nervous tissue consume only glucose in all metabolic states except for prolonged fast, where 2/3 of the brain will start to use ketone bodies, like we mentioned earlier about ketone bodies. And then last but not least, we have integrative metabolism, which is talking about our metabolic rates. So let's go over calorimetry, which is measures metabolic rates. So you can literally measure the heat a person gives off. And so in respiratory quotient, RQ, it estimates the composition of fuel that is actively consumed by the body. So this is a formula, CO2 produced over O2 consumed. So you can't see what someone is burning, but you can measure what goes in and what comes out. And so this is the logic. The whole logic is how oxygenated the fuel molecule already is. So then we can go back to regulatory hormones. So ghrelin will increase your appetite. Oxorexin will increase your appetite as well. So any sign, sound, taste, or smell of food might increase your appetite, and these are the hormones on it. Leptin will decrease it and be like, oh, you're full. But it doesn't actually do that as a signal. What it does is suppresses oxorexin right over here. So it will decrease your appetite. And then your body mass index is your mass over your height squared. So mass is always in, it doesn't write it, but mass is always in kg, kilograms, and then what else? The height is in meters squared. So meters is height. So those are the SI units that we're using. All right, a useful diagram is this metabolism overview, which we finally got to look at. So proteins, carbs, and then fats and lipids. And then you can see how this is a big overview of energy. Metabolism, I said, was all about energy, right? Energy processing. So how your body is going to make energy is basically, or stores energy, uses energy, makes energy, etc. And so this diagram can help a lot during studying. We already went through all of these other ones. The ones that we didn't go through as much are these final pathways here, but they're not as high yield, right? I mean, they're high yield, but they don't take as much information space, if that makes sense. So fatty acid synthesis, and then you have beta oxidation, which is important too. So don't think they're not high yield; they're all important. Urea cycle, and then we have the pentose phosphate pathway here. So these are all important here too. Also understand and memorize how not only they're connected, but what they're doing, why they're doing these things, and then what products you're getting out of it. Here are the energy products for beta oxidation. Okay. So that was a whole review of the entire biochem. So I hope that was somewhat helpful in some way, and it helped review for a lot of people. I hope that reminders of reviewing other subjects was important, was helpful, because sometimes biochem is just a lot of the subjects mixed into one big subject. So yeah, all right, that's enough.